1 2 3 चेक चेक चेक। हेलो, ठीक है अरिजीत जी? ठीक है, ठीक है, चलो।
में कि तुम्हारी आवाज नहीं जा रही कम, तो यहाँ पे दोनों बटन है। ठीक है, बोलो।
Good morning.
Geology.
A very good morning to all the dignitaries, guests, delegates, and all my colleagues. On behalf of THSTI, I Pragya, extend a very warm welcome to all of you for THSTI 13th Foundation Day celebrations. As a network organization linking many centers of excellence, THSTI, with its collective multidisciplinary expertise, aims to effectively enhance the quality of human life by integrating the culture of shared excellence in research, education, and translational knowledge with the entrepreneurial spirit to take the novel technologies into the public sphere. To make the celebrations memorable, a series of scientific events and educational events have been conducted over the past 2 days. As we start the formal Foundation Day celebration, we are fortunate that we have our respected dignitaries with us. I request Dr. Sushmita Chaudhary to deliver the welcome address.
Good morning to all of you. We have gathered here for celebrating our 13th Foundation Day at THSTI. First of all, on behalf of our Executive Director, Professor Garg, and from THSTI community, I would like to extend our heartiest welcome to Professor Vinod Paul, member of Niti Aayog. Sir, you have been a guiding star for THSTI since very long, since your AIMS days. And we are really honored to have you back here. And we are eagerly waiting to hear the words of wisdom from you. Our most sincere welcome to Professor Rajesh Gokhale, Secretary DBT and Chairperson of THSTI governing body. You have been a pioneer of translation, innovation, and entrepreneurship in India. We constantly look up for inspiration from you. Our warm welcome to Dr. Alka Sharma, Senior Advisor, DBT. Welcome, ma'am. And Dr. Jyoti Loghani, our dear Nodal Officer, and other dignitaries from DBT. Also, we sincerely welcome all our previous directors and previous secretaries of DBT who have joined us online today. A very warm welcome to Professor Sudhanshu Bhrati, Executive Director of RCB. And very sincere welcome to all our colleagues from RCB and other collaborating institutes. Finally, from Professor Pramod Garg and from the THSTI management, a very warm welcome to all of you present here and all of you who have joined us online today to celebrate our 13th Foundation Day. I hope you enjoyed today's celebrations. We would like to start with lighting the diya. I would request our esteemed guests and Professor Garg to light up the diya and mark initiation of the celebration. Thank you, sir. Now we would like to extend a green welcome to all of you. Shilpa, may I request Shilpa? Shilpa to Professor Paul. Nidhi to Professor Gokhale. Sanskriti to Professor Garg. Thank you, all three of you. I would now request Pragya to carry on with the rest of the program.
Thank you, ma'am. I request our esteemed director, Professor Garg, to deliver the 13th Foundation Day inaugural address and present the annual report. Please, sir.
Good morning, ladies and gentlemen. Respected Dr. Professor V.K. Paul, member Niti Aayog, and former head of the Department of Pediatrics at AIIMS, New Delhi. Honorable Secretary of DBT, Professor Dr. Rajesh Gokhale, Dr. Vrati, Director RCB, Dr. Alka Sharma, Dr. Logani, my esteemed faculty from THSTI and RCB, all the scientists, postdocs, PhD students, and officials. It's indeed a pleasure for me today to give the annual report and tell something about THSTI, what we have achieved over the last 1 year. Before I do that, I must say that, sir, it is indeed our honor to have you today. I know Professor Paul is extremely busy. In fact, this morning also, he took an online Zoom call for more than an hour. That shows his commitment to science, health science, and improving the health of the society. Even just before we came here, we were discussing how to improve the growth and development and mental development of small children. You know, constantly he's thinking about health science, how to improve health of the people. Many of you won't know that he established the super specialty of neonatology in this country. At AIIMS, the neonatology center was the WHO Center of Excellence, and he laid the foundation of neonatology across the country. People used not to believe that very small babies of less than 600-700 grams of weight who are delivered prematurely even before 32 weeks or even 28 weeks would survive. But at neonatology center in AIIMS, such babies used to survive. And that was a big achievement of the neonatology department. And you would know that he has done so much for policy and initially for the pediatric population, but later on, once because of his credentials, he was made the member of Niti Aayog, which for us is a moment of proud. And now you see that as member of the prestigious body, he has contributed again for health sciences, for improving the health. And I want to tell you that Ayushman Bharat scheme, which is the premier scheme of the Honorable Prime Minister of our country, Shri Narendra Modi ji, Dr. Paul was involved in development of that scheme right from the beginning. In fact, he shared with me that once that how they worked very hard and presented to the Prime Minister this scheme. And he was saying that, why didn't we start it earlier than this? And there are always hiccups, ups and downs in the scheme. But today, you know that so many hundreds and thousands of families are benefiting from that scheme, thanks to sir's effort. So thank you very much, sir, for accepting our invitation to grace the occasion. Professor Dr. Gokhale, currently has dual charge of DBT and CSR. He is extremely busy, I know that. Yet I insisted that he should come and grace the occasion. He was a bit reluctant because he has no time right now. And I know that. And I really felt a bit sorry when I insisted that he should come. only because the institution needs you, because the faculty, the scientists, the students feel inspired by the presence of people like you. You know, if an eminent scientist becomes the leader of the group, it inspires people. It tells you that this is how science must be done. This is how you should move forward. And people like him can guide the institutions because he has done it himself. He has seen it himself. So that is the reason I requested that you must be here. And thank you very much for accepting the invitation and being here. I know it will mean a lot. It means a lot for us, a little bit hardship for you, but thank you once again. So today is the 13th Foundation Day of THSTI, established way back in 2009 under the visionary leadership of Dr. M.K. Bhan, who was the secretary at that time. And Professor Paul actually has been involved in this process right from the beginning. So I call it an oasis for translational research. People often ask, what is translational research? And, you know, when you have a research project, you have what comes out of it usually is a publication. The moment you have a product coming out of that, it becomes translation because that product is immediately used for patient's benefit, be a diagnostic test, be a therapeutic molecule. So this is what translation is all about. And it is both from bench to bedside and from bedsides to bench. When you have a problem, you go back to the bench and find a solution for that. The COVID crisis that we have seen for last 2 years has actually brought biotechnology and health sciences to the front. Now everybody, right from the highest authorities, the policymakers, and the public understands the importance of biotechnology and health sciences, whether it is molecular testing, whether it is antibody kit development, be it vaccine development or viral sequencing. Everything requires biotechnology and health sciences. And actually, India has done extremely well in all these areas. Last month, under the leadership of Dr. Rajesh Gokhale, DBT and BIRAC, inaugurated the India Biotech Startup Conclave. And the Honorable Prime Minister actually mentioned that Indian biotech industry is worth $80 billion as of today, with more than 70,000 startups. And India is going to be soon among the top 10 countries, thanks to the effort of all the scientists and faculty involved and entrepreneurs involved in the biotech industry. And therein comes the role of institutions like THSTI, which is a catalyst and enabler of translation in the research ecosystem in our country. We conduct theme-based research programs, prioritizing public health problems, mainly in the area of infections and immunology, mother and child health, non-communicable diseases, and diagnostics. And these are ably supported by major facilities that we have: BSL-3 facility, biorepository, data center, bioassay lab, animal facility, and also something which is rather unique is Clinical Development Service Agency, kind of academic CRO conducting various trials. So it is a unique institution in its own sense, combining basic biology, clinical research, diagnostics, and also clinical trials. So I'll just briefly touch upon the research that is being conducted here, facilities that we have, latest developments, our achievements, and what our future plans are. So one of the flagship programs of THSTI is in the area of maternal and child health, led by Dr. Chinjin Bhatnagar, interdisciplinary team dedicated to maternal and child health research. And we all know about this Garbhini program, which has successfully been completed of more than 6 years now. And it is looking at various factors, both antenatal and natal, for the prematurity. Multiple papers have come out. In fact, they have now come up with newer guidelines for gestational weight gain, also looking at air pollution, how it affects pregnancy, and also omics-driven biomarker discovery and validation in large cohort of these patients. Also, intervention trials are ongoing. One of them is adjuvant zinc therapy for young infant sepsis. They published a paper on kangaroo mother care in the prestigious NEJM. And also now the Action 3 trial is ongoing with regard to the use of steroids. And many papers have come out, I think more than some 25 or so, in very good journals. And I think India is making, globally making a mark in this area. And now, thanks to DBT, the part 2 of the Garbani has been sanctioned, it is ongoing. This is looking at growth and development from birth to 2 years of age. Many objectives there, but primarily looking at prenatal and natal exposure in terms of growth and development of the child and guidance for intervention in future. Influenza is another area where THSTI is doing very well. Again, thanks to DBT, we have 3 major grants on flu and influenza vaccine development, and very happy that now ferret facility has been fully established. We have ordered the ferrets now, and anytime soon they will be arriving for research on influenza vaccine and ferret models. Tuberculosis is again a major research program at THSTI looking at disease biology, therapeutics, and vaccine. CRISPR-Cas9 is one tool that has been used primarily by Nishit and his group, and it is being used around the globe now. Ramandeep and Amit Pandey are looking at design development of major therapeutic molecules, newer molecules, looking at identification of molecules through screening of FDA-approved drugs, through target-based screening and phenotype-based screening. And I'm sure there are 6 or 7 molecules that are showing very high potential to be taken up for the next phase. And of course, there are multiple publications through these efforts. Microbiome is now all over the place in health and disease, and everybody talks about microbiome. And at THSTI, it is being led by Bhaptosh, a major program. We are looking at microbiome in different sites of the body and association with different disease states. And now there is a potential where we can actually manipulate the microbiota and go for therapeutic effect. Immunology core is again major area here, which is led by Amit. And this is immunobiology of infection, non-infectious disease being looked at. T cell biology, particular Th9 cells biology is being investigated, and they are doing both fundamental applied research. And I think during COVID also, the cellular immune response assays developed by the immunology core actually helped understand the cellular responses to vaccines, and they came very handy for all the vaccine development in the last 2 years. Non-alcoholic fatty liver disease is something that is again being investigated both for its pathophysiology and also drug discovery through different approaches, targeted and non-targeted approaches. In fact, we now have a grant from BIRAC and Dabur India to look at the effect of certain Ayurvedic products in a very scientific manner. So Ayurvedic products definitely might have some benefit, but the only problem always is whether they have been scientifically tested or not. I mean, just can't say it is very effective, we have to test it. So I'm very happy that there is now a very scientifically driven program for this. In fact, last month we organized a one-day symposium on non-alcoholic fatty liver disease, which was attended by more than 300 delegates, and we had more than 25 faculty for this very successful program. And yesterday we had another one-day symposium as part of our Foundation Day celebration on inflammation, attended by more than 500 delegates, and there were more than 20 faculty. It was a very successful symposium on inflammation. CDSA has been conducting 17 trials. Recently, actually, 8 have just finished. This is led by Nitya as the officer, faculty in charge of CDSA, and I think CDSA is doing a good job, very, very good job. I have earlier talked about COVID-19 research at THSTI, but might be a good idea to just to recapitulate that we have been working in the development of vaccines, animal models to test therapeutics and also vaccine, immunogenicity and cellular assays, biorepository, and cohort development. And I'm happy, sir, more than 31,000 patients or vaccine cohorts have been developed in different areas, whether it is those who have been infected with COVID, those who have been vaccinated, those who have hybrid immunity, those who have Delta, those who have Omicron. So all kinds of cohorts are now there. And based on that, last year, we published during the height of Delta that how Covishield is effective in 63% of patients, of people preventing the Delta infection. We are also fortunately part of INSACOG now, thanks again to DBT support. We have done more than 1,000 sequencing, maybe now actually some 13 or 1400. And we have recently identified the BA.5 in one of the patients, and the same virus has been cultured also. THSTI is the coordinating center for this large hospital network study where 14 clinical sites have been linked with sequencing sites. I think it's kind of a unique study. I have not seen any such study in other parts of the world where each site is linked with the sequencing sites. And now we have more than 3,100 patients' data that have been prospectively collected. And I think about 40% of them have been sequenced. The reason some of them have not been sequenced is either the viral load was low or the sample wasn't good, but we have enough data to say that what is the likelihood of an outcome of Delta infection, because we started when the Delta was still prevalent. What is the outcome of Omicron, Omicron B.2 or Omicron even 0.5? So, and now again, I'd like to thank DBT that this study is being extended by another 1 year so that we continuously keep looking and do surveillance for this. In fact, sir, in India, THSTI was the first lab to culture Omicron virus, and ours were the second report. First was published in Lancet, second from our country was published in Lancet eBiomedicine, where we showed how the plasma from infected and vaccinated people is suboptimal in its efficacy or neutralization against the Omicron virus. For vaccine development, of course, THSTI has contributed immensely for vaccine in the country, Sputnik 5, Zydus, Biological E, and also now we have, in partnership with Panacea, indigenous multi-epitope vaccine against SARS-CoV-2, and actually animal challenge models are ongoing. And we have a large influenza research group of many scientists, and the group is leading the effort of indigenous vaccine. I'm very happy to announce, sir, that THSTI along with industry partner Panacea got a funding of $12.5 million from CEPI to develop a new novel highly immunogenic pan-beta coronavirus vaccine. We have received the grant, and this grant is going, the work has already started, and we hope in next 2 to 3 years, we should have some good outcome of this. We also developing some neutralizing antibodies against SARS-CoV-2, and two scientist groups are working on this. One of the antibodies actually shown very good results. This was a rat and a mice antibody. This has now been humanized, and this is part of a grant from BMGF of $1.5 million for studying, developing cohorts, studying their responses, and also developing antibodies. So we have 2 good antibody candidates, and I hope that, and one of them is actually working against Omicron also. So this is being taken up, and there have been many, many publications on COVID-19 in the last 2 years, more than 50 publications in very good journals from THSTI. Among the new facilities, this is a new BSL-3 facility, which was inaugurated by the Honorable Minister last year. This is a shared facility between RCB and THSTI, and I'm very happy that now the facility is actually functional. The good part of this facility is it is modular. We have 4 modules, so you can start 1, 2, or 3, 4 modules. This will be very, very cost effective, and this is now open to other institutions and also to industry if they want to use this important facility. Some of the new equipment that you installed, now we have a complete platform for proteomics and metabolomics. Complete solution is there. We have sequencing with us, and we have a Schrodinger for new drug discovery through the in silico approach. And we are soon going to acquire this year a next generation sequencing platform, so that it is not just adding capacity, it is also for training and our PhD and postdocs in all these techniques, because we can easily outsource such a thing, which may be more cost effective in the beginning. But I think long run, what is required is that institution like this should have their own equipment so that our students can be trained on that. We have started a collaboration with DNDi India for dengue therapeutics, and Dr. Guru Prasad is working on dengue, and now we are testing many therapeutic molecules, mainly the repurposed drugs against dengue in both cellular and animal models. We are going to start a School of Diagnostic Innovation by Design. In fact, we received the in-principle approval from DBT last year, sir, but because of some reasons, the funding hasn't yet come, and we hope that we have all the infrastructure ready with us, and we hope that this should be operational this year itself. Another important thing that THSTI is starting is MSc program in clinical research, and this has been now registered with RCB, and thanks to Dr. Vrati's efforts, their board has approved it, and we have the faculty in place, and this year we are starting a 2-year program in MSc clinical research, which I think will go a long way in capacity building in clinical trials in our country. The new facilities that we have planned this year, and again, I would like to thank the Secretary, Dr. Gokhale, for approving many of our projects. For example, through INSAPI program, we are now developing a new vaccine development design center within this campus. We have taken input from industry also, because now we will have an antigen come out of the center, which is ready to go to the industry for further development. So this will be very important. And as mentioned last year, we have all the clearances for a medical research center. In fact, 2 clearances were required. One is zoning planning and the other is environmental clearance. Fortunately, both have been cleared at the highest authority in Haryana government. We are just waiting for their formal letter. And once we have these formal letters, we will be in a position to start the construction work. Also, a new research building for THSTI was approved 3 years back, but again, because of COVID, there was a delay. But now we have, again, DBT has sanctioned money for that. We have land, and we are going to establish a new research building with newer facilities. Upgradation of animal facilities, again, has been approved in principle by DBT. We were waiting for some, final clearance, and I hope within the next week or 2, we should have the final letter of approval. We have the money with us. They have already given money, and I think the plan is ready, and we should be able to do have a world-class facility here. Some of the achievements here: publications about 64, 5 patents were filed and granted. These are some of the very good publications. 5 scientists have secured faculty position in various institutions in the country. I think this speaks about THSTI as a very good institution. Last year's grant was cut to about 44 crores, sir, but this year we have been sanctioned 70 crores. Again, I'd like to thank the Secretary and the financial advisor for giving us adequate amount of funding. We received total of 136 crores funding last year, and I'm very happy to say that THSTI could generate almost about 11 crores through various revenues, mainly through collaborative work and research work. So that's a sizable amount, and I hope this trend continues, because this was partly because of the COVID work, but I hope that this will continue in future as well. Moving forward, sir, the only challenge we face currently is that our number of faculty members is less, and I requested our secretary this morning that we should move the file, and he was very positive, and he said, you just move and hopefully this will be sanctioned. So this is something that's on the top of my agenda now. And we promise, sir, to make THSTI a flagship institution of DBT, which DBT under your leadership will be proud to have. Thank you very much for your kind attention.
Thank you, sir, for your inspirational speech and summarizing the THSTI's achievement and future plans. As a part of scientific events, a colloquium was organized for the 4th and 5th year PhD students of the Institute. I now request Ms. Upasna Madan to present her research work. Ms. Upasna has been awarded the best oral presentation in the colloquium held on 13th of this month.
और पॉइंटर कैसे हेलो नहीं आएगा
Slides. Good morning, everyone. I'm Upasna Madan, PhD student under the supervision of Dr. Amit Awasthi. Today, I'm going to give Foundation Day colloquium presentation. The title of my presentation is Role of FOXO1 in Immune Tolerance. A brief introduction. There are two subsets of the CD4 positive T helper cells, which are pro-inflammatory T cells and anti-inflammatory T cells. Pro-inflammatory T cells include IL-17 producing cells, which are TH17, IFN gamma producing cells, which are TH1, and IL-4 producing T cells, which are TH2. And the anti-inflammatory T cells include FOXP3 positive T cells, which are T regulatory cells, and IL-10 producing T cells, which are type 1 regulatory T cells called TR1. Although inflammation is required for fighting infections, but whenever there is hyperinflammation, it leads to the pathology. So in the autoimmune diseases, the major effector T cell population which leads to the pathology are the TH1 and the TH17 cells. They causes the tissue inflammation, and the role of the anti-inflammatory T cells is to inhibit the effector function of the pro-inflammatory T cells and resolve the inflammation. Therefore, a relative balance between the pro-inflammatory and anti-inflammatory T cells is required to maintain a homeostasis state. In my PhD research work, we are majorly focusing on a transcription factor, FOXO1. So FOXO1 is regulated by a highly conserved PI3-AKT kinase pathway. Whenever there is activation of the PI3 kinase, it leads to the phosphorylation of the AKT. AKT in a phosphorylated form, it moves inside the nucleus and leads to the phosphorylation of the FOXO. FOXO in the phosphorylated form is moved outside the nucleus into the cytosol, and it leads to the inhibition of its transcriptional activity. For FOXO to be activated, FOXO1 should be in unphosphorylated form present inside the nucleus so that it can regulate the expression of its target genes. The FOXO1 is regulating a variety of the diverse biological processes like metabolism, cell cycle arrest, apoptosis, and DNA repair. Apart from these processes, FOXO1 has been reported to play a role in immune cells. Like T cells, they require FOXO1 to populate the peripheral lymphoid organs. FOXO1 regulates the L-selectin, CCR7, and IL-7 receptor molecules, which in turn regulate the homing, migration, and survival of the naive T cells. Also, FOXO1 is known to regulate the T helper cell differentiation, specifically the T regulatory cell development and function. Although FOXO1 has been reported to regulate naive T cells, homing and survival, and regulatory T cell differentiation and function, it is still not clear the role of the FOXO1 in effector T cell generation and function, and the role of the FOXO1 in the pathogenesis of autoimmune diseases. Now, to study the definitive role of the FOXO1 specifically in the T cells, we have generated a FOXO1 conditional knockout mice. For the generation of this mice, we have procured FOXO1 flox mice and CD4K transgenic mice from the Jackson Laboratory, USA. FOXO1 flox mice is having the LOXP site surrounding the FOXO1 gene, and CD4K transgenic mice is having the CD4 Cre recombinase present, which is specifically expressed in the CD4 positive T cells. We have bred and mated these mice in the small animal facility, THSTI, and we have generated CD4 Cre FOXO1 flox mice. So this mice is having the FOXO1 deletion specifically in the CD4 cells, because Cre recombinase will be expressed only in the CD4 cells, while the rest cells in the mice, they are FOXO1 positive. This is the agarose gel picture where we used to genotype the FOXO1 mice using standard PCR-based protocols available on the Jacksons. It could be seen that this is the wild type allele. This band is of the FOXO1 flox mice, mutant mice, and the third band is of the heterozygous mice. It is having one wild type allele and one flox allele. This is the agarose gel image of the CD4 genotyping. This is the transgene band. After the generation of this FOXO1 conditional knockout mice, we have performed a cohort study for over a year where we have understood the natural pathogenesis in this knockout mice. For over a year, we have taken wild type mice and the FOXO1 knockout mice, and we have taken the weight of the mice weekly. And we observed that the wild type mice, they keep on increasing their body weight, but FOXO knockout mice, they do not show much increase in their body weight. And there is a significant difference in the body weight of wild type mice and the knockout mice. Also, these mice, within 6 to 8 months of the age, they start to showing signs of the spontaneous encephalitis. As it could be seen that they are having a hind limb paralysis and there is a loss in the tail tone. Because they are showing the sign of the inflammation of the brain and the spinal cord, we have done a HE staining of the brain and Luxol fast-bleed stain of the spinal cord in the 1-year-old wild type and the knockout mice. And we observed there is a huge inflammation and huge infiltration of the immune cells in the brain, and there is an increased demyelination of the spinal cord of the knockout mice. So this observation are pointing that FOXO1 is very important to prevent the autoimmunity. We also did hematoxylin and eosin staining of the different organs of the 1-year-old wild type and the knockout mice. And we observed that the pancreas is having huge inflammation and infiltration of the immune cells. Similarly, in the liver, the knockout mice are having huge inflammation. And in the lungs also, we observed they are having interstitial pneumonia. There is infiltration of immune cells and there is bronchitis. Colon histopath images also showed that the knockout mice are having infiltration of immune cells and inflammation. Because we are observing huge inflammation in different non-lymphoid organs of the knockout mice, we looked at the surface marker, which is CD69 expression. CD69 is expressed on the activated T cells, and we observed that the knockout mice, CD4 T cells in the mesenteric lymph nodes, peripheral lymph nodes, and spleen, they are showing a higher expression of the CD69. We also did a surface profiling of CD44 and CD62L. CD62L is a marker which is present on the naive T cells, and CD44 is a marker present on the memory T cells. We observed that knockout mice is having a very less population of the naive T cells and a very high population of the activated T cells, which are CD44 high cells. We also performed the cytokine profiling of effector cytokines IL-gamma and IL-17 and anti-inflammatory cytokine IL-10. We observed that CD4 cells of the mesenteric lymph node, peripheral lymph nodes, and spleen in the FOXO knockout mice, they are showing higher expression of the IL-gamma and very high expression of the IL-17, which also confirm our finding that because there is a high expression of the effector cytokines, they are having huge inflammation. We also did the cytokine profiling of the lamina-propria lymphocytes isolated from the small intestine of the wild type and the knockout mice. And we observed that there is a very clear distinct population which is IL-10 positive and CD4 positive in the lamina propria of the wild type mice, but this population is absent in the knockout mice. Also, they are having a very high population of the IL-gamma positive, IL-17 positive, and IL-gamma IL-17 double positive T cells. So this double positive T cell population is a major effector cell population causing the pathogenesis in the autoimmune diseases. Now to confirm our findings that FOXO1 mutant T cells, they are showing more differentiation towards effector T cells. We performed in vitro differentiation using the splenocytes of the wild type mice in the presence of the FOXO relocator. FOXO relocator is a drug that will activate the FOXO1. We observed that the activated T cells are activated in the presence of the FOXO relocator. There is increase in the expression of the IL-10. We performed in vitro differentiation of the TH1 cells in the presence of the FOXO relocator, and we observed there is increase in the expression of the IL-10, and there is an increase in the expression of the IL-gamma IL-10 double positive T cells, which are anti-inflammatory T cells. We also performed the in vitro differentiation of the TH17 cells in the presence of the FOXO relocator, and we observed when the FOXO1 is activated, there is inhibition of the IL-17 positive T cells, and there is increase in the IL-10 positive T cells. So this shows that when FOXO1 is activated, the previously affected T cell population can acquire an anti-inflammatory T cell effect. Now to confirm if the FOXO relocator can be used in treatment of the autoimmune diseases. And to confirm our findings in vivo, we performed DSS colitis model. So it is an experimental model of colitis used commonly in the labs to perform colitis in mice. So dextran sodium sulfate is a compound that causes the inflammation inside the colon and leads to the hyperactivation of the immune cells. We performed a study where we have taken the group 1 mice which are on the 2% DSS in their drinking water for 5 days, and then they were kept on water so that we could see the recovery from the colitis. And then another group of mice, FOXO relocator was administered intraperitoneally 15 mg per kg on day 0, day 1, day 3, day 5, day 7, and day 8. Weight of the mice was taken every day, and disease progression was followed. We observed that the DSS mice, they're showing a significant decrease in their body weight, while the FOXO relocator mice, they did not show any decrease in their body weight and they regained their body weight. So there was a significant difference in the colitis disease between the DSS mice and the mice which were given FOXO relocator. In the DSS group, there was a shrinkage of the colon length, while in the FOXO relocator group, there was no shrinkage in the colon length. And the HE staining revealed that the DSS group, there is a huge inflammation and infiltration of immune cells, while in the FOXO relocator, the colon is intact and the villi structures are also intact. Now to look at the cytokine profiling in these groups, we observe that the relocator is having a huge anti-inflammatory T cell population in the lamina propria lymphocytes, and there is a reduced population of the IFN gamma and IL-17. And similarly, in the mesenteric lymph nodes of the FOXO relocator, there is a reduced population of the effector cytokines, which are IFN gamma and IL-17. Now to give a summary of my presentation is that when the FOXO1 is present in the CD4 T cells, there is a balance between the IL-10, which is an anti-inflammatory cytokine, and the pro-inflammatory cytokines, which are IFN gamma and IL-17, and hence a homeostatic condition is maintained between the effector T cells and regulatory T cells, and it creates a situation of homeostasis and immune tolerance. While when FOXO1 is mutated or deleted in the CD4 T cells, there is a disbalance. There is very less anti-inflammatory cytokine and a huge population of the pro-inflammatory cytokines, which leads to a disbalance between effector T cells and regulatory T cells, and the person becomes more prone to the autoimmune diseases, which are systemic lupus erythematosus, psoriasis, autoimmune diseases of the colon, inflammatory bowel disease, and the arthritis. I would like to acknowledge my PhD supervisor, Dr. Amit Awasthi, for teaching me, guiding me, and his constant support throughout the work. I would like to acknowledge my doctoral committee members, Dr. Vineet Ahuja from AIIMS and Dr. Amit Kumar Pandey for their valuable discussions during the DC meetings. I would like to acknowledge the THSTI FACS facility, Dr. Deepak Kumar Rathwar for guiding me in the FACS. I would like to thank my lab members for their constant support. I would like to acknowledge Small Animal House facility at THSTI and ILBS in New Delhi for the histopathological imaging. I would like to acknowledge the funding facilities, TST SERB and the UGC for the funding of this project. I would also like to acknowledge friends and family. Thank you.
Upasna, we wish you all the best for your future endeavors. We are honored to have with us the Secretary of Department of Biotechnology, Government of India, our Nodal Ministry. And also, he is the Chairman of THSTI governing body, Professor Rajesh Gokhale. Professor Gokhale joined NIIS faculty after his postdoctoral training at Stanford University. He is well known for his metabolic studies, diversity in the pathogen. His research focus over many years has been on elucidation of interplay between metabolic reprogramming and immunity in the context of tuberculosis. He was the director of IGIB, Institute of Genomics and Integrative Biology. He has several honors to his credit, and to mention a few are the fellowships of all the 3 science academies and the Shanti Swarup Bhatnagar Prize for Science and Technology. It's our pleasure to welcome you, sir, for the foundation address.
Close this.
Thank you very much for inviting me here. आप सबको मेरा नमस्कार. Honorable member of Niti Aayog, Dr. Paul, Executive Director of THSTI, Professor Pramod Garg, other distinguished guests from THSTI, DBT, RCB, other neighboring institutions online, my dear students and researchers, postdocs, fellow staff of THSTI, and ladies and gentlemen. It's indeed an honor for me to be participating in this 13th Foundation Day lectures, Foundation Day of THSTI, for several reasons. Even though, Pramod has rightly said I was very reluctant to come, there are really three main reasons why I've always wanted to come, but it was very hard to come actually. I mean, one obviously is the dual charge of executive authority makes life very, very complex actually. If you in an executive stance, you really always are the bottleneck to do whatever everybody else wanting to do actually. And this poses even more responsibility on you to be able to facilitate the two large organizations that we're looking at actually. But as I said, THSTI has a special role in my mind actually for primarily because I've been associated with its inception since the time Dr. Bhan, in fact, started this conceptualizing and romanticizing this idea of setting up THSTI. I think I distinctly remember several meetings that he used to hold in the evenings, actually, that I used to participate along with Dr. Martha from the HST Institute and several of others. Dr. Rati is not here, but he was part of it. And it used to be, every week we used to hold this meeting and it used to go on for a couple of hours. And as you know, Dr. Bhan, being a very, very involved person, would always engage himself in every single small activities that's part of it. And it's always good to see, what has come out of it, right, actually. And you always really enjoy the feelings of time and investment of energy that people have put in who had visions to be able to carry out these activities. So obviously, coming to THSTI always remembers me of one of my mentor, Professor Bhan, who's been, incredibly important in my life and career. The second really is that being part of the Delhi culture, NCR culture, there are lots of friends and colleagues whom I know here actually. So it's always good to come and see them actually. And, the nice smiles and exciting faces to look at. It always gives you the pleasure of, rather than sitting in your single room and, carrying out activities that are important. The third and foremost, in fact, is this campus provides immense opportunities for future, actually. If you really look at from a perspective of what one needs to grow, what one needs to aspire, and what SNT needs to deliver in terms of its societal responsibilities, this campus really holds all the promise, actually. And really, that's as Dr. Pramod very articulately suggested, how wonderful stuff all of you have been doing, actually. I mean, how can we really enhance it to make a tremendous amount of impact? Because this is, the India is a very, very large country, actually, right? And what one needs to do is really have an impact. To make a real impact, one needs to have larger number of faculty members, larger number of value chain additions, even enormously capable activities. Many of them, as you rightly have started, actually, is really what made me come here and say, let me spend some time and to really speak my mind out, actually. I think reluctance on my part has been primarily because, as I believe that as a Secretary Department of Biotechnology, my job profile is very, very different. Of course, this is a part of a responsibility as well, but it's really new job for me as well. I mean, it's about 8 months that I've been at DBT now, and I'm learning a lot of things as at different places. You have different things to learn. And I clearly realize that my role is actually at 3 different levels, to be very frank, if I look at it, at investment, in policy, and in regulation, actually. And all these 3, in fact, embodies what DBT organizations would do it in future and would carry out activities, because the umbrella of this is really what makes stronger vision and perception of what one needs to do. Either it's got to do with individuals, it got to do with communities, or it got to do with societies and community involvement. All of these becomes a very, very powerful mechanism of successful stories ahead, actually. So I think, I was just, in the morning, I was just sitting and thinking about, 10 years back when THSTI, about 13 or 15 years back now, and, I got a little bit disconnected with THSTI because that's the time when I moved to IGIB as a director. and I had different responsibility to look at. But I think I still recollect a day when Dr. Bhan, in fact, held a very, very long afternoon meeting to evening with Professor Eric Lander and Professor David Baltimore, who had visited, and there was no online meetings. So it was rather involved meeting at NII. This was held at NII, where we had this long discussion over where should be the THSTI, what should be the contour of THSTI, how should THSTI develop into an organization that can carry out these activities. And tremendous amount of learning at that younger age for me, actually, believe me. I mean, this is about 2008, and I'm probably about 8 years into my research and activities. But it's so very important to, in fact, think through larger issues and programs and policies very early in your career that really changes your perspective of what one wants to deliver, how one wants to evolve, and how one wants to actually, in fact, manifest the way you look at come back. You know, sitting in this chair now, if I look at why do we, you know, for example, we can ask ourselves, why do we practice science? I don't know how many of you asked this question. And the simplest answer for many of you could be inquisitiveness, right? And there's nothing wrong with that. I mean, I have a profession, I'm very inquisitive, and I'm trying to look at it. From a government point of view, why would government invest in science, actually? And if you look at from the government point of view, it is always what I call the progression of promise, right? That the science progression will enhance the quality of people and human life in the society perspective, actually. And I think the whole idea of Department of Biotechnology is really to bridge these two things, is to bridge the inquisitiveness of scientists and the governmental aspect of the perspective of, you know, what I call progress of promise, or, you know, power of promise, whatever you can call, actually, because if science is a power of promise, because eventually only science and technological breakthroughs can really make changes and can bring differences to the world. So really what I believe is it's a combination of imagination and progression that one can really merge together to be able to catalyze enormous amount of activities that one is looking forward to. And I think to do this, it is very, very important that all of you come together as a team and convert this place into the best possible institution in about next 5 years to come, actually. And I think that's what the message that I wanted to give you before I, just say a couple of, just to say that, this is very, very important for organizations to have larger perception, to understand what their founders, in fact, had the vision for, what kind of ideas they wanted to look at. And it's always continuously important to reverberate through that thoughts and ideas to look at. In fact, if you look at from a government point of view, I mean, it's a tremendously active government and large number of things that are happening that many of you may be partly aware of or maybe fully aware of it, which in fact also involves what is called rationalization of autonomous institutions, as many of you would hear about it actually. And I will briefly give you a perspective about it. Why would you really want autonomous bodies to merge and they make into a bigger entity actually? And it's very important from a point of view, look at one of the first and most important, I mean, right now there are 14 autonomous institutes in Department of Biotechnology. Each one of them have their own governance structure, of which I am the governing body chair. And therefore what I means is that I am the chairman of the governing bodies of all these institutions. That means I need to worry about how the governance structures of all these autonomous bodies are functionally being communicated across to people, because that's the whole idea of governing bodies are. If you just imagine sitting through these 14 governing bodies and trying to imagine and think through, can we come up with practices and solutions that are in the best interest of all organizations? It can be quite a demanding, challenging perspective actually to look at. And so therefore, to bring in efficiency into the system, mergers into larger organizations, in fact, can bring in tremendous amount of value actually. Because these governing bodies are not just an aesthetic purpose, but in fact, their role has to be in a very clearly defined manner, the way business entities function in their board meetings and carry out these activities. Actually. Also, the other problem is if you look at internationally, THSTI, if anybody comes and visit THSTI and asks a question, how many number of faculty members do you have on your board? And if you really look at, if you compare it with any organization across, I mean, you will always, the person will say, oh, that's really true. You are such a small institute and have so many things, activities going around here. So clearly the impact is also very small. For example, I was talking to people, there are two different challenges that India faces, which is rather interesting to think of. John Hopkins team was visiting DBT and we were discussing about neuroscience program and they wanted to and then I asked him how many people do they have in neuroscience in John Hopkins, and this is about 500 faculties actually in that one single building. And we're not even 500 in Department of Biotechnology actually, to be very frank. The whole of DBT constitutes of less than 378 people right now in terms of scientific community to look at. And then you say $80 billion industry that we want to create, and our ambition is $150 billion industry actually. So clearly our footprint is very, very small. On the other hand, people from Denmark and other places, Novo Nordisk and all these people would come and tell you, oh, this is the way we have done our stuff. This is the way we bought diabetes and insulin and in all this part of the country. And their problems are very different. And if you really look at their problems, they're looking at a problem that is even smaller than the total population of Noida actually. So the demography of complexities, on one side, the number of people that cater to scientific community, and on the second side, the value chain that you want to add is so skewed and so extreme that we are always under tremendous pressure to, in fact, carry out innovative solutions and technological parameterization to really make tremendous amount of impact. And I think it's very important, therefore, we understand that how do we make it impactful? And I think that's where I think Foundation Days and developing these ideas are very, very critical to important to sit together and think together as a team how we can deliver, what we can do, and how can we carry out these important tasks. Finally, I would just say that I think I was just a couple of days back, and sometimes some things just get set me. I was just reading a poem actually, and somebody was reciting this poem, and I thought it's very, very incredibly what I call communicative of the thoughts and ideas that sometimes people talked about. And this was in Hindi. Let me try and communicate. I'm not one of the best person to memorize things, but just in the end to say, this is a very beautifully written. It says ki it talks about sun and says Surya aap kitne mahan ho karke, and nadiya parvat pahad sab kuch sukha dete ho aur usse whatever kara dete ho, but aap fir bhi garib ka pasina nahi sukha pate ho. And I think what that poet is trying to say is incredibly amazing actually to look at. Is very often we get fixed in the fact that we are very big, we have this tremendous opportunity, we can do many things, but very simple tasks you can't achieve despite doing that actually. And they are of tremendous, incredible value. So one of my last point to tell is really develop empathy for people whom we are responsible for. And you will in fact make a big nation, great nation that we all really look forward to actually. I think that's what are my last messages. But finally, I would like to really thank Dr. Paul. He being honorable member at NITI Aayog has been of tremendous value to me. He's been a great guidance for me actually. And whenever I have a problem, I pick up my phone call and call him, and is ever so gentle mannered and very, very nice and polite. And thank you, sir, for all your help. It's been a great learning for me for last 8 months, and I hope we all can together make a big difference. Again, thank you, Dr. Garg, for inviting me, and real pleasure to be here amongst all my friends and colleagues.
Insisted him to come over here. That's the reason. You need such an inspirational speech, and this is so very important for the institution. Thank you very much once again.
Thank you, Professor, for your enlightening words. We are profusely elated to introduce our chief guest, Professor Vinod Kumar Paul. Professor Paul is one of the most renowned pediatrician and neonatologist in the country and globally. He completed his under and postgraduate medical training from AIIMS, after which he joined as a faculty in the Department of Pediatrics in his alma mater. He chaired the department for over 10 years. After completing 4 illustrious decades at AIIMS, he was nominated by Government of India to NITI Aayog in August 2017, where he led the health, nutrition, and the HRD verticals. He has played a pivotal role in the formulation of key initiatives such as Ayushman Bharat, PMJ, Ayushman Bharat Health and Wellness Central Scheme, and Poshan Abhiyan. In the wake of COVID-19 pandemic, Professor Paul chairs the National Task Force on COVID-19 and the Empowered Group on Medical Emergency Management Plan. He also serves as chairman of the National Expert Group on Vaccine Administration for COVID-19. Professor Paul has featured among the world's top 2% scientists in the field of pediatrics. He is a fellow of all the three science and medical academies of the country. It's our pleasure to have you, sir, with us. I would now request you to please deliver the keynote address of the Foundation Day.
So I have the permission to speak from here. Good afternoon, Namaste, Abhinandan. Most respected Dr. Rajesh Gokhleji, Secretary DBT, respected Dr. Pramod Garg, Executive Director, THSTI, Dr. Bharati, Dr. Alka Sharma, Dr. Shinjini Bhatnagar, distinguished faculty of THSTI, students, scholars, postdocs, colleagues. First and foremost, my deep gratitude for the kind invitation, Dr. Garg and the faculty, for many reasons, and I'll come to that. Thank you very much for this opportunity to be here on the 13th annual day, Foundation Day of THSTI. THSTI is very close to my heart, and I'm so grateful that you have shared affection and special attention to me today. As Dr. Gokhale said, in my own way, perhaps to a lesser extent than Dr. Gokhale, been part of the conversations with late Dr. M.K. Bhan on THSTI composition, contours, systems, vision, expectations, objectives from time to time. Today we all must remember the visionary leader in science, Professor Maharaj Kishan Bhan. I'm so honored to be in this hall, which you have enshrined in the name of Dr. Bhan, my friend, philosopher, guide. We miss him every day, and I feel that he is here amongst us today. Over the years, this institution has reached adolescence, so congratulations, and that calls for an applause. Journey of 12 years is not small, and yet it is not that large. It is time to celebrate our achievements along a difficult path. Establishing an institution is not easy, and early years, sir, are not easy. And 12 years journey has produced so much of science and science products that we are proud. But it is also a moment for charting the next phase of our journey through adolescence to adulthood. So in some sense, this is a very special moment. And I'm sure you all will discuss where we are and where we should be 25 years from now when India celebrates 100 years of its independence. Already, you have served the cause of humanity. You are jewel in the crown of India in science, for sure. And there's more to come. I'm truly impressed with your achievements, a plethora of achievements, and the span of work that's being done. And I echo what Dr. Gokhale said, small size and yet big achievements. And that augurs so well for future. I particularly would like to congratulate Upasana for her work, and big applause once again to her. tuberculosis, maternal and child health, NAFLD, hospital network, and above all, huge work on SARS-CoV-2, its vaccine, its pathogenesis. Very quiet work. Dr. Garg, I particularly want to congratulate you and thank you and your team for the quiet work that you did for almost every vaccine that this nation produced, the assay that you did, the network that was used, and the science that you produced to help them. Not so visible to people, but I know how your teams work day and night to produce the data on efficacy for vaccine A, vaccine B, and vaccine C. Thank you so much for being a leader in ensuring that high quality objective science powers the development of vaccines by the companies. Thank you for doing it. Sir, I can see that here science stands tall, but science also is connected to humanity. Father of the nation said, science without humanity is a sin. What I see here is science for humanity and for underlined. So thank you for delivering on that. And Dr. Gokhale also mentioned that we need to create science and knowledge, but it should be seen, it should be meant for, in our state of our nation's development, for the people, for the quality of life of the people. And you clearly, practically every theme that you picked up, which you presented, sir, shows that connect. And I think that was in the DNA of THSTI in some sense. That was the DNA, this translation. It is very beautifully, you said, research moving to publication, and then it moves to product, becomes a translational science. So science for humanity, and I again would like to congratulate the entire team for remaining on that path, within that bandwidth, very effectively. Thank you for doing it. Bench to bed is the classical translational science paradigm we are taught, we are told. But I was also told at some point, again by Dr. Bhan, that there is a valley of death. Solutions that are sitting on the benches don't reach the beds, and beyond beds to the homes of people, the communities, and so on. So what you're trying to do is to create a bridge from bench to the bed and beyond. What we realize when you take this journey, that it is actually not one valley, but many valleys. Valley of regulatory processes, sir, that we were trying to sort out, valley of funding, valley of industrial partners, the GMPs that you have to do, and valley of getting a product into the market to be owned, to be placed in the public health system, for use in the public health system. Lots and lots of valleys. So one request I have and a suggestion is that we create connects with potential people who are part of those facilitations, which will overcome the dips that you may encounter. I hope the students are taught and the faculty is informed about regulatory processes, how to build public-private partnership, how to ignite a startup, how to be able to reach the industry, how to create funding. I hope that that conversation, which is not difficult to bring to this cluster because it is all over, but it should be exposed to the young people from the beginning, rather than getting stuck as it has happened I'm sure in your case, in my case, from time to time. So I think it's one thing to create a potential product, but it is another thing to actually make sure that the product also reaches where it should reach. And therefore, one paradigm of fusion that I like to see would be to those pressure points which make life difficult, but to inform them and to see how to navigate that journey, which will eventually ensue. I'd also like to see my second suggestion, more collaborations. I did hear a little bit less on the collaborations. Collaborations not only with AIMS, which is an obvious one, which of course I saw in Upasana's papers and I know other work that's being done, but also other institutions and also not so famous institutions, because there are very bright people, sir, in the not so famous institutions. Sometimes the focus on the very visible is right, but sometimes we miss creating a stimulus of right kind in the institutions that are, at least where the students are there, who are bright and can get attracted. So more collaborations, and then reaching out to say medical colleges around us, to other universities around us. One opportunity that you may like to think to provide, and I see plenty of work going on, and I think it's possible, is to get science students and medical students as a fellowship, perhaps continuously. हमेशा ही यहाँ पे 20 लोग घूम रहे हों जो जगह जगह से आए हैं, not only from the premier institutions, but also from other institutions who are just feeling the potential of this great place and may get attracted to science and use. They are interns, that's it. They are here, they are fellows. Of course, you can do more of them in summertime or whatever light time you have. But I think that would be a good service for us to do, to create young people, to stimulate the young people from various institutions. Another obvious fourth suggestion I have is the connect with the startup culture. Maybe it is strong and you have, the lot of pathways, and within the DBT system itself, we are doing a lot. One suggestion is that you may like to make a connect with the Atal Innovation Mission, which is housed in the Niti Aayog, where a lot of young people are producing technologies in another world and looking forward to connect. So it's one area where one platform, which I think will help bring in more vibrance to the work that you're doing. Some today, but even much more as we move along. When I see the products that are coming from there, 40% at least are related to health. Often these are AI type technologies, but then there are also hardcore products. So your biodesign center, but also from here, taking products to that side and connecting with the young startup, young people who are, emerging into the system through that system would be useful. That's my other suggestion. I like to remind all of you a couple of things which Dr. Bhan used to say. So two things. He would say, and I quote, take up high risk, high impact areas. I want you all to be thinking big from the founder of this institution. High risk, take risk. But if that risk is being taken for something which will eventually have high impact, go for it. Absolutely go for it. Because this institution, the executive director just tells me that you have practically all the facilities that the best centers have, minus maybe 6%, 8%. That gap will be bridged. You have everything, practically everything. So pick up on the high impact, high risk areas and work in those areas, because that is where you would make a difference. And to get that, he says, and I quote again, keep going, do not give up, success will come, unquote. Keep going. Don't give up, and success will come. So this is one thought I wanted to share with you, because I heard this and the proxies of this so often from Professor Bhan. Two other suggestions, and this is my address primarily to students and scientists. One, that excellence is not about what you will do great on a particular day. Excellence is a habit. I quote here none other than Aristotle, and you may know this quotation, but that's my favorite, and that's one message that I have for you. Aristotle says, and I quote, "We are what we do repeatedly. We are what we do repeatedly." Excellence then is not an act but a habit. We are what we do repeatedly. Excellence then is not an act but a habit. You'll do something great today. You produce something beautiful. You write something beautiful today. Your experiment is perfect today. That's fine. To show to Dr. Kokle or to your director, बहुत अच्छा हो रहा है. लेकिन क्या यही चीज हम रोज कर रहे हैं क्या? उसी फाइनेंस से कर रहे हैं, उसी एसओपी से कर रहे हैं. और क्लिनिकल साइड में मेरे पास ज्यादा एग्जांपल हैं कि वही आप पेशेंट के लिए जो कार्ड पे लिख रहे हो, कोई देख रहा है तो अच्छा लिख रहे हो, समझा रहे हो, क्या वो रोज करते हो? क्या हर बार ही करते हो? जो आप एक बार ही करके बेस्ट दिखाते हो वो आपका पोटेंशियल है. जो आप रोज करते हो वो आप असल में हो. तो अगर आप असल में एक्सीलेंस रिप्रेजेंट करते हो, then you must perform to your best capability on a daily basis as a habit. And I urge you that you follow this principle. That's what we need, because if India has to surpass our neighbor and beyond, it's all about excellence, and it's all about excellence every day. So that's one message. My second message to you all is to truly think big. We are passing through these 25 years, what the Honorable Prime Minister says, Azadi ka Amrit Kaal. Alag sochna padega. To go beyond what this generation, all of us have done. You have more opportunities, you have more resources, and you can leapfrog several things that have been done by other people. You can pick up areas which are in early stage where you can compete other people out. Sometimes difficult to compete people out here. There you can only do by jumping through technology and other things. But you enter an area which is live, which is not too late from here. Think about those areas. So think big, dream big, डरना नहीं चाहिए. किसी ने सोचा था कि इतना अच्छा क्लस्टर बन सकता है फरीदाबाद में? बाय द वे, ये क्लस्टर जो है वो डॉक्टर भान का सबसे प्यारा क्लस्टर है. उन्होंने और भी बनाए हैं, पर यही उनका सबसे ज्यादा उनकी भावना इसके ऊपर थी. दैट आई नो. उसकी बाकियों की बात कभी कभी करते थे, इसकी रोज करते थे. सो आप बड़ा सोचो. ये मेरा एक आपसे एक अनुरोध होगा. And here I like to again quote one of my favorite quote or favorite belief, favorite guidance. And this comes from an African lady, Ellen Johnson Sirleaf, who was the first elected woman president in Africa, in any country. She was president of Liberia. And she says something very sums up. In fact, it echoes Dr. Bhan's philosophy beautifully in a way. But let me give the quotation to you. And I quote. She says, and listen to it carefully at least once, "The size of your dreams must always exceed your current capacity to achieve them." ध्यान से सुनो इसको. The size of your dreams must always exceed your current capacity to achieve them. If your dreams do not scare you, they are not big enough. Unquote. The size of your dreams must always exceed your current capacity to achieve them. If your dreams do not scare you, they are not big enough. अब तो यंग हो, ये मत सोचो कि इतना ही है आज तो मैं इसके अंदर क्या कर लूं, ये नहीं सोचो. डॉक्टर भान भी कहते थे, जब विजन सेट कर रहे हो ना, तो रिसोर्सेज की बात मत करा करो. वो बात की सोचा जाए, आने दो. मैं वही रिफ्लेक्शन इसमें देखता हूं और साथ में कहता हूं, आज इतना है यही मत सोचो, ये बढ़ेगा और होगा. तो ड्रीम इसके अंदर फिट मत करो. ड्रीम देश के लिए सोच के हम कहां ले जा सकते हैं उसके ऊपर फिट करो. ये मेरी आपसे गुजारिश होगी, आप तो लीड करोगे। हमारे देश की डेवलपमेंट में साइंस ही तो हमें लेके जाएगी। साइंस ही हमें क्लाइमेट चेंज से हमें बचाएगी, हमें सोल्यूशन देके। इतने बड़े चैलेंजेस जो हैं वो साइंस, कुछ भी कहिए आप, वो साइंस ही हमें उनसे उबारेगी। Having said therefore, size of your dreams must not be through the lens of today's resources. कभी इतना नहीं सोचो, बढ़ जाते हैं, अपॉर्चुनिटीज बढ़ जाते हैं, तो ड्रीम ऐसा होना चाहिए कि बिल्कुल अलग से हो। और एक ड्रीम मैं देता हूं कि अगले 25 साल में हमारे देश में इस अमृत काल में 2047 तक एक नोबेल लॉरिएट, एक नोबेल प्राइज आएगा, आ सकता है। एंड could that Nobel Prize be associated with this institution? जहाँ का स्टूडेंट हो, यहाँ का काम हुआ, ये संभव हो सकता है। क्या हम उस तरफ जा सकते हैं? तो एक चैलेंज है, ये मेरा ड्रीम है जो मुझे भी स्केयर कर रहा है। अब मेरे गूज बम्स हो रहे हैं, करके दिखाते हैं ना, ऐसी चीज पकड़ते हैं। सब कुछ है आपके पास, कोलैबोरेशंस हैं, रिसोर्सेज हैं, आइडियाज को करो ना। वैसे बदले कोई सोच सकता था कि हम 9 महीने में एक वैक्सीन बन सकता है? कोई मानता ही नहीं था, बन गया ना। एक नहीं बना, कितने बन गए? थर्मोस्टेबल mRNA, वो इंट्राडर्मल भी बन जाए, बताओ। सब देख रहे हैं कि देश ने क्या करके दिखा दिया। कोई नहीं सोच सकता। परसों तक हम 200 करोड़ वैक्सीन लगा चुके हैं। वॉच दैट डे, वॉच दैट स्पेस। सो एक ड्रीम छोड़ रहा हूं यहाँ पे इस महफिल में। इतने एस्टीम्ड साइंटिस्ट हैं मेरे आसपास, लाइफ दिया है इंडिया की साइंस के लिए, छोड़ के आए बाहर, कितना काम किया। क्लिनिशियन है वो जो क्लिनिशियन का जोश होता है, जुनून होता है, उसको छोड़ के साइंस को प्रमोट कर रहे हैं। टीम है यहाँ। और ह्यूमन हेल्थ के लिए या एनिमल हेल्थ के लिए इस प्लेनेट पे हम इतना बड़ा क्या काम कर सकते हैं। सो दैट इज a dream that I leave here. I like you to think about, spread the good word, and be proud of your institution. Stand for the values that make an institution a great institution. शुरू की लीडरशिप की एक जिम्मेवारी है, जहां लीडरशिप बैठी हुई, उसको कहना चाहता हूं कि you set the pace and the future of the institution. You set the standards. You set the culture. तो 12 साल होने पे एक ये भी इंट्रोस्पेक्ट करने का जरूरत है कि क्या उस कल्चर में अगर हिला हो रहा है, यंगस्टर आ रहे हैं नहीं फिट हो रहे, उसमें कैसे उसको ठीक करना है। ये चैलेंज आता है। एंड आई हैव सीन इट एट एम्स। हम सब एम्स में उस कल्चर की ही कोशिश करते रहे जो हमने अपने सीनियर्स में देखा। कहीं सक्सीड हुए, कहीं कमी रही, कहीं हो सकता हमने उसमें बढ़ोतरी भी करी अपने तरीके से। सो एट 12 years दिस इज वन थिंग। और ये गवर्नमेंट के लिए वहां से नहीं आएगा ये चीज, ये हमारी भावना, हमने करके दिखाना है कि कहीं इस कल्चर में जो शुरुआत हुई या जिस विजन से ये काम शुरू हुआ, एंड द ऑडिटोरियम इन विच वी आर सिटिंग टुडे, द सोल ऑफ द फाउंडर अराउंड अस, लेट अस रिफ्लेक्ट एंड सी हाउ वी कैन बूस्ट द कल्चर further and align it with the culture of the best institutions. वो वैल्यूज परवेड होनी चाहिए। तो अपनी कोर वैल्यूज को फिर अलाइन करते हैं आज on this 13th annual day. I'm so happy, so delighted to be here because I have a special feeling. I have benefited in my own work with the early phase of this great institution. It's a great vision. Like to see many, many products reaching the bedside, reaching public health delivery systems. I'm very, very proud to be here. Thank you so very much.
Thank you very much, sir, for these encouraging, inspiring words. Truly appreciate it. Thank you.
Thank you, sir, for your words of wisdom and encouragement. Honorable Minister of State IC of the Ministry of Science and Technology and Earth Sciences, Dr. Jitendra Singh ji, wasn't able to join us today, but has kindly sent us a message for the THSTI family. Honorable Dr. Jitendra Singh is an elected member of the Lok Sabha for the second consecutive term from the Udhampur constituency in his native place, Jammu. He graduated from Stanley Medical College, Chennai in 1978 and completed his MD medicine from Government Medical College, Jammu to become an eminent diabetologist and endocrinologist. As a physician politician, he serves across various portfolios as Union Minister of State, Ministry of Development of Northeastern Region, Ministry of State Prime Minister's Office, Ministry of State Ministry of Personal Public Grievances and Pension, Minister of State Department of Atomic Energy, and Minister of State Independent Charge in the Ministry of Science and Technology and Ministry of Earth Sciences. I request our esteemed director, sir, to please read out the message.
Thank you, Honorable Minister. Actually, was planning to come here. In fact, till day before yesterday, we were hoping that he'll be here, but then obviously he has a very, very busy schedule and had to go out of time. Yet he was kind enough to send a message to THSTI, and I'll read it out now. It's quote. In recent times, large investments in biomedical sciences are required for better health of our citizens. With progress and improved understanding of human pathogen genomes and emergence of personalized medicine, cost-effective and indigenous solutions become crucial and require bringing together a broader expert pool of talent across disciplines. Integrating different areas of science under one roof is critical to explore and find quicker solutions for solving the unmet needs. Also, collaboration between academia and industry is important to bring forth health innovations. Numerous such efforts have been initiated by THSTI. The Government of India, under the leadership of our Prime Minister, Shri Narendra Modi ji, gives great importance to innovation for Atmanirbhar Bharat, Made in India and Skill India. THSTI has significantly contributed in this value chain, especially in the last few years. The future belongs to technology-driven economy, and THSTI is critically placed for building innovations ecosystem in the country. I congratulate the faculty and students of THSTI on their Foundation Day. Unquote. Thank you very much.
I request Director Sir and Professor Sunjani Bhatnagar to felicitate our esteemed guest with their small token of appreciation and memento. Ma'am, I would request you to please come over to the dais. We would also like to felicitate our biggest supporters, the DBT officials. And also to our RCB director, Professor Sudhanshu Vati. And to the deputy official. Achievement and success are not by accident. Dedication and commitment serve as a vital link in the growth of any institution. So, as a symbol of the Institute appreciation for dedication and loyalty of many staff members and students, we commence the award ceremony. These awards are distributed every year during the Foundation Day celebration under various categories. The first award in the category is Dr. M.K. Bhan Group Award for the most impactful collaboration is awarded to the GSIR project. The PI of the project is Dr. Pramod Garg and co-PI is Dr. Jayant Bhattacharya. Global Immunology and Immune Sequencing for Epidemic Responses, GISER, is envisioned as a collection of sites that can combine local pathogen sequencing, surveillance, and clinical epidemiology with local immunological understanding and tools. The project entitled GISER South Asia has been awarded to THSTI by Bill and Melinda Gates Foundation, which aims to increase the in-country capacity for evolution of SARS-CoV variants of concern in South Asia. It brings together the pathogen sequencing capacity, local human subjects research, clinical epidemiology, and local virology and immunology expertise to allow in-country scientists to rapidly evaluate new variants for immune escape and cross-protection. The GISER project consists of the clinical team led by Dr. Sinjini Bhatnagar, with its members namely Dr. Nitya Wadhwa, Dr. Ram, Dr. Pallavi Chitrapal, Dr. Deepika, Dr. Mudita, and Kondi and Dr. Kondi, and the other members are Dr. Jayanta Bhattacharya, Dr. Gaurav Batra, Dr. Sweeti Samal, Dr. Tripti Srivastav, and Dr. Shailendra Mani. I request the team to please come over the dais to receive the award. The second award for the faculty for the best published paper goes to Dr. Amit Avasti. Dr. Avasti's research work has been published in various journals during the year 2021. The award is for papers published in the Lancet Infectious Disease having an impact factor of 25.05. The next award goes for a PhD student for the best published paper goes to Dr. Ramachandran Thiruvangadam. Dr. Dr. Deepika would be receiving this. Dr. Ram is being awarded for the best published paper in the Lancet Infectious Diseases, having impact factor of 25.071. Award for a PhD student for the 5 years all-round performance goes to Mr. Rajdeep Dalal. Mr. Rajdeep established animal models for IBD and identified molecular pathways and preclinical validation of new therapeutic targets which regulate intestinal inflammation. In addition to his PhD thesis work, he contributed significantly for various other research projects as well as has published various papers. The next award for a faculty for overall contribution to the institutional development during the previous financial year goes to Dr. Nitya Wadhwa. Her primary research interest in finding solutions for neonatal and early life morbidity and mortality. Dr. Wadhwa has built and led multi-site, multidisciplinary research teams that are capable of performing research to the most exacting. She has contributed to interventional trials in the maternal and child health domains that can improve preterm birth outcomes and many others to her count. The next award is for the administrative staff for overall contribution to the institutional development during the previous financial year. It is jointly shared by Mr. Deepak Badoni and Mr. Manjeet Kumar. Mr. Deepak successfully processed multiple tenders on GEM and GBP portal for the bulk purchase of laboratory consumables. He was instrumental in implementation of Central Store Facility module on ETHSTI portal to facilitate scientists' regular requirements to avoid the time delay. Dr. Manjeet has contributed in managing the construction projects of THSTI, which includes Office of Connectivity and BSL-3. He is responsible for all construction activities, including providing technical inputs for methodologies and construction and coordination with site management activities. The next category of award is
because he gives awards, for administrative staff. So big round for Mr. Santo.
the next award for a technical staff for overall contribution to the institutional development during the previous financial year is again jointly shared by Mr. Pradipta Jana and Mr. Sakib Kidwai. Dr. Jana is absent. I think somebody from the lab would be accepting the prize on his behalf. Dr. Jana has tremendously contribution in the COVID-19 genome sequencing and bacterial whole genome sequencing. THSTI has complete genome sequencing of more than 1,000 COVID-19 positive samples and has made a substantial contribution for the same. Mr. Sakib has associated with the institute since 2009. He has also contributed in setting up the new BSL-3 labs of THSTI. He is also in charge of the library of the institute. He looks after the functioning of the IDRF labs at THSTI. Moving ahead to the prizes of the scientific events that were conducted in the past 2 days. We had poster presentation competition in 2 categories. The first category was the PhD students and project associate. The first prize in this category goes to Ms. Joshna Dandotia. The second prize goes to Mr. Shivam Kumar. And the third prize goes to Ms. Sneh Bajpai. The second category too was MK Fund Fellows and Research Associates. Among this, the first prize goes to Dr. Chetali Singhal. The second prize goes to Dr. Ashish Kumar Agrahari. And the third goes to Dr. Pranav Tripathi. We had another category 3 for the best PhD student in the second year that goes to Ms. Samriddhi Mehta. Coming to the prizes of the colloquium competition, the first prize goes to Ms. Upasna Madan. The second prize goes to Ann Sugandha. And the third prize goes to Ms. Manisha Singh. Manisha, you have been awarded as the third prize in your colloquium. Please come to the dais. She just entered the auditorium. With this, we have approached the end of the first half of the Foundation Day celebration. I would request Dr. Krishna Mohan Atmakuri to please come to the dais for his vote of thanks. First part.
On behalf of the Executive Director, the faculty, the scientists, students and staff of THSTI, I sincerely thank Dr. Jitendra Singh Ji, the Honorable Minister for Science and Technology and the Minister for Earth Sciences, and the President of THSTI Society for his presidential message and encouraging words to mark our Foundation Day. I profoundly thank our Chief Guest, Professor V.K. Paul, member of NITI Aayog, for not only sparing his valuable time despite his busy schedule and joining us in our Foundation Day celebration, but also delivering the keynote address on how to move forward, dream big, and take maximum risks. Your inspiring, insightful, and encouraging words means a lot to us. Thank you very much, sir. I warmly thank Professor Rajesh Gokhale, Secretary DBT and Chairperson of the governing body, for delivering again a very insightful, encouraging words and address, the Foundation Day address, and his best wishes for the constant support and the direction towards THSTI. Thank you very much. I warmly thank Dr. Alka Sharma, ex-nodal officer for THSTI, and now the senior advisor to DBT and MD Barak, and for her constant support towards THSTI development. I warmly thank Professor
Sudhanshu Rathi, Executive Director, RCB, for joining in the celebrations and his constant support. I also thank Dr. Jyoti Lagani, the current Nodal Officer for DBT, Dr. Vaishnav, and Dr. Vandana from DBT for being here. I thank all the members of the THSTI Society, the governing body members, the Scientific Advisory Committee members, the Scientific Review Committee members, and the Financial Committee members for their constant guidance and support. I take this opportunity to thank all the officials, the scientists, and the program coordinators of the DBT for their constant support. I also thank all the state officials who joined us today and extending their support towards the development of THSTI. I thank all the collaborators and the funding bodies who work with us in the growth of the science. I thank our Executive Director for his constant direction and vision towards success of the Institute in achieving its goals and vision as quickly as possible. Now I invite you all for lunch, which is organized in the Dome area. Request you all to assemble back at the auditorium by 2:15. Post lunch, we will have a quiz competition, followed by which the entertainment program and an exciting talk by Dr. Richard Haynes on the therapeutics for SARS-CoV-2 and beyond. Look forward to seeing you all by 2:15. Thank you.
हेलो जल्दी जाओगे जल्दी पाओगे आगे वीआईपी जल्दी वापस आ जाना खाना आके वापस आ जाना बोलो हेलो बाहर ही हो। अरे नहीं, यहीं पर। यहाँ के बच्चे कहाँ हैं?
Shabnam, and Samriddhi.
So for the quiz competition, I would like to call teams on stage. Team A, Bela Desai and team. Team B, Akshay Binayak and team. Team C, Daisal Talukdar and team. Team D, Roshan Kumar and team.
टीम बी
B है, B है.
ये टीम ए है शायद बेला देसाई ये हाँ ये टीम ए ये टीम ए है हाँ हाँ मैं बता दूँ वो है टीम टीम सी
जैगम सर पिपलीज कौन सी चलाने मुझे दे देंगे तो मैं उनको फीड दे दूंगा फिर फीड ही चलेगा पहले चला देना है मैं चला दूंगा
इसका फर्स्ट पेज जो है ना उसमें रूल्स लिखा हुआ है तो रूल्स खोल लीजिएगा ताकि
मैं बोल
चारों को सांगता फर्स्ट देंगे एक ही प्राइस देंगे कोई कंपटीशन
ठीक है सर फीड दे दिया
फर्स्ट आना
ओके अभी सिर्फ नेक्स्ट करना है आपको पीपीटी ऑपरेट कर लोगे ना सर आप ठीक
हाँ वो बोले थे इस साइड रखो हाँ वहाँ से दिख रहा है सबको दिख रहा है कोई तो क्वेश्चन आंसर अभी है सर अभी राउंड हाँ बहुत ज़्यादा यूज करेक्ट नहीं वो ठीक है क्वेश्चन आंसर देने लायक गलत हो गया
वो रेगुलेशन को अच्छे से पढ़ानी पड़ेगी
So the rules for the quiz are displayed over here. I would request everyone to read it carefully. So rules. No. So the rules for the quiz are displayed over here. I would request everyone to read it carefully. So 3 questions will be asked to each team per round. Round 1 will have 4 options. Round 2 will have audio clips, images and logos. Round 3 has 5 questions in total, and it's a buzzer round. The first team to press the buzzer will get the opportunity to answer. The teams will be given 30 seconds to respond. An unanswered question will not be passed to another team, it will pass on to the audience. Each correct answer carries 10 points, and there will be no negative marking.
Jay. Okay, it's not working now, sir. Kuch. Hey, Bhagwan.
I would request every team member to introduce their members. So starting with Group A, please introduce yourself. Yes.
Yeah, hi, I'm Bela. I'm a program manager in MCH.
Hi, I'm Dr. Srivatsan. I'm project coordinator in Geyser and I work at MCH.
Hello everyone, I'm Mudita Gosai, PhD student, MCH.
Hello everyone, I am Akshay Binaike and I am a PhD student at THSTI.
Hello everyone, this is Tarini Vora. I'm a project associate in Dr. Vasti's lab.
Hello everyone, I'm Eman. I'm also project associate in Dr. Vasti's lab.
Hello everyone, I'm Dr. Daisy, working as a RA in Dr. Bhabatosh Das lab. And my friend Shantanu and Medha, they are both from Dr. Das lab.
Hello everyone, myself Roshan Kumar, working as lab technician in Dr. Bhabatosh Das lab.
Good afternoon, everyone. This is Shushmita Bakshi, working as a project associate in Dr. Bhabatosh Das lab.
Good afternoon, everyone. This is Shruti Lal, and I also work as a project associate in Dr. Bhabatosh Das lab.
Okay, so probably we are done with the introduction. So herewith, I would like to welcome you all on the behalf of quiz team. I am Samriddhi, and I hope you enjoy this game as much as we enjoyed preparing it for you. So basically, quiz is a competition of mind, intellect, interest, power, and joy. And so without wasting much of your time, since we have already introduced the teams and we have already told you the rules, I would quickly want to brief you that this had been a very pretty journey where we had initially 15 teams and they were all superb. There was a neck-to-neck competition between all of them, and we got these 4 really intelligent and intellectual teams out of all 15 of them. And so I would herewith start the competition and call Shabnam on the dice so that she can read out the questions.
So starting with first round, which is MCQ round. The first question is for Group A. As per the World Population Prospect 2022 report, India is projected to surpass China as the world's most populous country in which year? Okay, I will repeat the question. As per the World Population Prospects 2022 report, India is projected to surpass China as the world's most populous country in which year?
B.
So, sorry, wrong answer. It's 2023. So, next question for Group B. Which of the following academies is responsible for fostering the development of dance, drama, and music in India? Options A, National School of Drama. Option B, Sangeet Academy. Option C, Sahitya Academy. Option D, Lalit Kala Academy.
Option C, Sahitya Academy.
Wrong answer. It's Sangeet Academy. So, question 3 for Group C. Who among the following are an Indian Olympic archer and Padma Shri winner? Option A, Kidambi Shrikant. Option B, Bajrang Punia. Option C, Balbir Singh Dosanjh. Option D, Limba Ram.
And the time is up. The question is passed to the audience. Can anybody want to try? Do anybody want to try? Oh, okay.
Correct answer.
Right. That's the right answer. Thank you, audience. So it's good to see that the audience is also participating, and it's really upsetting to see that the quiz participants are not answering the right. Try your level best, Baba.
And please, audience, अपना level for control रखिए. Question number 4 for Group D. I hope you answer this. The academic center of the Indian Space Research Organization is set up in which state? Option A, Kerala. Option D, Karnataka. Option C, Andhra Pradesh. Option D, Tamil Nadu.
The audience is requested to please remain quiet. Let the participants answer.
It will be passed on to you. If
Option B, Karnataka.
Correct.
Right.
So next question for Group A. Which classical dance is named after the village it was originated from? A, Mohiniyattam. B, Bharatnatyam. C, Kuchipuri. D, Kathakali.
D, Kathakali.
Sorry, wrong. Audience? Sorry? Yes, correct. It's Kuchipuri.
I think the guess went wrong.
Okay, next question for Group B. Ministry of Electronics and IT has integrated the India Meteorological Department IMD services on which application? Option A, Shashank. Option B, Umang. Option C, Shakti. Option D, MyGov.
Your timer has started. Your time is running out.
Sorry? No, wrong. It's audience. Umang, correct answer.
This team.
Yeah, audience are answering every question. Okay, next question for Group C. Indian men's hockey team defeated which of the following teams in Tokyo Olympics 2020 for the bronze medal? Sorry. Option A, Belgium. Option B, Australia. Option C, Germany. Option D, Holland.
A, Belgium. Belgium.
Wrong. Audience? Correct. Germany.
The audience are displaying the power of Ekta mein Shakti hoti hai. So everybody is like compiling their voices and their voices are coming out to be one.
Okay, next question for Group D. I hope you answer this. Question is, who among the following declared Women's Cricketer of the Year for 2021? Option A, Indian opener Smriti Mandhana. Option B, Tammy Beaumont of England. C, Lizelle Lee of South Africa. And D, Gabby Lewis of Ireland.
Option A, Smriti Mandhana.
Correct. So this is last round for this MCQ part. So level up. Okay, Team A. Which city has been given the status of World Heritage City by UNESCO in July 2019? Option A, Jaipur. Option B, Jodhpur. Option C, Bikaner. Option D, Ajmer.
Okay. D, Ajmer.
Sorry?
Ajmer, D.
No, wrong. Audience?
There are mixed voices. Let's say which part. This part, what does you say?
This part? It's Jaipur, correct. Okay, next question for Team B. Please answer this, guys. How many seasons of the TV show Friends were there?
Shanti.
It's for group.
Aapka time aayega.
10.
Correct. 10. Okay, Team C. What does airbag used for safety of car drivers contain? Option A, sodium bicarbonate. Option B, sodium azide. Option C, sodium nitrite. Option D, sodium peroxide.
Audience, please.
Option C, sodium nitrite.
Sorry?
Sodium nitrite. C.
Sodium nitrite, no.
No, it's wrong. Let's hear it for the audience.
Okay, wait.
Wing wise.
I'm not able to do it.
Acha, it's okay.
Wait.
Sorry.
So who's saying option A? Who's saying option B? C or C? It's option B. It's option B. Okay, next and last question. Ghumura is an ancient folk dance that originated in which of the following states? Orissa. Wait.
Audience.
Options.
C.
Wait, let me read the options.
Audience, we do have
It's C, Rajasthan.
Sure. It's wrong. It's audience. Who is saying Orissa?
Orissa is the right answer. So, Ghumar,
it's not
Ghumar for Rajasthan. It's not Ghumra.
It's Ghumra.
So there's a change.
We thought it's a different name.
So final score for after round 1 is?
Team D leading 20 points.
And the audience has please requested to remain quiet because there would be a mini quiz competition for you. When the team fails to answer it, it will be passed to you. So we will be passing it wing wise. So it's really very requested to you that please remain quiet so that we can enjoy the competition silently and yet enjoyable.
Okay, so after round 1, marks are Team D is leading with 20, Team B is second with 10 points, and Team C, please make some points.
And also we need to avoid the bias in the quiz competition. So another thing, we will be starting with the second round, that is audio and the visual competition.
So for second round, audio and visual, first we have 4 logos. We will show you the images of logos and you will have to identify the brand, okay? So starting with Team A, this is the logo.
Schrodinger.
Correct. Correct. It's Schrodinger. For Team B, this is the logo.
Fast track.
Correct. Please, audience, let them answer first. Okay, for Team C, this is the logo.
Airbnb.
Correct. Airbnb. Last logo for Team D.
Manas, I can see you waving your hand. Let's wait for the timer to end.
Pass.
Pass. Okay. Manas, the question has been passed to you.
Correct, Sun Pharma. Okay, so next we have some audios. You'll have to recognize the band or movies it is. Okay, we'll have some audio. So for Team A, this is the...
You're insecure, don't know what for. You're turning heads when you walk through the door. Don't need makeup to cover up. Being the way that you are is enough. Everyone else in the room.
One Direction. One Direction.
Correct. One Direction. Team B. So you have to recognize the movie name. And here it is.
दिलों में तुम अपनी बेताबियां लेके चल रहे हो तो जिंदा हो नजर में ख्वाबों की बिजलियां.
Can you answer this?
जिंदगी ना मिलेगी दोबारा.
No, it's Bhag Milkha Bhag.
Akshay expression is saying he want to listen it again.
That's what we are choosing because I also got confused with the answer. Okay, so there is a confusion whether it's Bhag Milka Bhag or Zindagi Na Milegi Dobara.
दिलों में तुम अपनी बेताबियां लेके चल रहे हो तो जिंदा हो.
Okay, for this we will confirm this. Okay, in 5 minutes. Okay, we will confirm this. We will confirm this. Me. We are not making.
तुम अपनी बेताबियां लेके चल रहे हो तो जिंदा हो नजर में ख्वाबों की बिजलियां लेके चल रहे हो तो जिंदा हो. हवा के झोंकों के जैसे आजाद रहना सीखो. तुम एक दरिया के जैसे.
Okay, fine. Sorry, it's Zindagi Na Milegi Dobara.
See, ये confusions रखे जाते हैं ताकि सारे लोग पता लगे कि सब participate कर रहे हैं. We knew it. It's जिंदगी ना मिलेगी दोबारा.
Okay. Team C, recognize the band.
You don't gotta go to work.
It's Dua Lipa and
Sorry?
Dua Lipa.
Who in the audience?
It's, you need to tell the band name. Correct. Okay, so it's audience. Sorry? Correct. Fifth Harmony. Fifth Harmony. Fifth Harmony. Okay, team.
It's a band of four.
Team D, please recognize the movie name.
डर गए? किस बात का डर है तुमको? किस बात का डर है? अरे खोने के लिए है क्या तुम्हारे पास? कुछ है? क्या है?
बोलना नहीं है, बोलना नहीं है.
Let them answer, guys.
Hold the excitement. जिस हिसाब से डिस्कशन चल रहा है लगता है पास ही हो जाएगा. सुनाई नहीं दिया? Let's play it again.
डर गए? किस बात का डर है तुमको? किस बात का डर है? अरे खोने के लिए है क्या तुम्हारे पास? कुछ है? क्या है?
It's time up, guys. You want to answer?
It's from Hrithik Roshan movie Super 30.
Okay, correct.
So Super 30 was a movie where it was based on a real biopic and जहाँ पे एक IIT का professor teaches underprivileged students and they also crack the NEET, right?
So last round for second part. So you have to recognize the images and tell whether which movie or which is it associated with. So this is for group A. Guess the band.
Yeah, Jackson 5.
Correct. So group B. Recognize the movie from this shot. Correct.
Piku.
Team C, please recognize the city with this image.
Audience, आप टीम डी हो क्या या सी हो? ये तो मुझे मुंडिया ऐसे हिलती हुई दिख रही है. Discussion is going on.
What?
Jaipur.
Correct. It's Jaipur. Okay, team D, please recognize the movie.
Ravan.
Correct.
I think the most excited person in the whole OD is Manis. या तो उसके हाथ ऐसे है, या फिर he's saying yes. I will tell
You
them.
should have been here. आपको यहाँ पे होना चाहिए ना.
He has always been, I guess.
So we are done with round 2 and the scores are Ravan.
देखो दो राउंड के बाद में टीम बी और टीम डी 40 पॉइंट्स और टीम ए 30 पॉइंट और टीम सी 20 पॉइंट्स.
So after round 2, team B and team D are with 40 points are at first position. Team A with 30 as the second position and team C with 20 is last.
But we answered the last question.
Sorry?
We answered the last question. We're still at 20?
40, I said 40.
I thought we were at 20.
टीम बी और डी 40 पॉइंट्स.
It's team B and the D that is on 40 points.
Correct. So we are starting with the round 3, which is buzzer round. Okay, see, you guys have buzzer. So whoever knows the answer, please press the buzzer fast. So the first question. Okay, so first question. This. Team A.
Lakshya Sen, Kadambi Srikanth, Team India won the thing, but they were the singles on the thing.
Yeah, correct. Next question is this.
Panchmahal is located in
Where? Team B. Correct. Fatehpur Sikri. Okay, next question. Who was known as the king of Bollywood music? Team A. Team A.
R.D. Burman.
Correct. R.D. Burman. Okay, next question. Chlorophyll is an actually of Team B. Let me read the question first.
It's a size. No?
But should I read the question? The answer is magnesium, and the question is chlorophyll is a naturally occurring chelate compound in which central metal is magnesium. So last question. Which Indian sportsperson won the World Games Athlete of the Year 2021 honor? They press it first.
Neeraj Chopra.
Sorry?
Neeraj Chopra.
No. Team B? No.
Okay.
Anyone from the audience?
वहाँ पे D ने बजाया था ऐसे.
अच्छा, D group D, sorry.
C. अच्छा, C ने बजाया था. अच्छा, everybody is reading the question and then answering.
Options तो है नहीं.
It's Hima Das.
MC Mary Kom.
हाँ, एक काम करते हैं, सारे स्पोर्ट्समैन के नाम ले लेते हैं और पास द राउंड खेलते रहते हैं.
So anyone from audience? No?
No. अब नहीं
No, Manas ji.
बोलेंगे.
It's let me answer. It's P.R. Srijesh. So with this, we are done with the round 3. So let's see the scores and see.
First team B, 60 marks. Second team. Next.
Okay, let me announce the result. Group B is the winner with 60 points. Group A is second with 50 points. Group D is at the third position with 40 points. And Group C is at fourth position with 20 points. So I would request the winner team Group B to come on stage. Group B winner Akshavinayaka, Tarini and Evan. Congratulations.
The prize distributions would be held at the end of the session. Thank you so much for joining in.
Thank you, everyone. Thank you, everyone.
Thank you. I thank all the quiz competition organizers for such a knowledgeable session. We really enjoyed it through. Moving to the next part. Moving into the next schedule, I am now handing over the stage and the podium to Dr. Yogita Adlakha for the entertainment session.
Good afternoon, everyone. I, Yogita Adlakha, Sub Research Scientist, welcome you all to the entertainment program. Dance is an epic form of gesture that portray emotions, joy, feeling, and love. According to Indian legend, the gods invented dance. For example, in Hindu mythology, a dance by Shiva creates and destroys the universe. Thus, dance tradition is thousands of years old and plays an important part in our Indian culture. And one of the most popular classical dance style of India is Bharatnatyam, which is the oldest dance form of India, and it was nurtured in temples and courts of southern India since ancient times. Our young researchers are also not devoid of art. Besides performing exemplary research, they are also talented dancers, singers, and actors. So here we present our first performer of the day. Put your hands together for Miss Shushmita, who will mesmerize you with her Bharatnatyam dance performance.
When I start.
मेरे ढोलना सुन मेरे प्यार की धुन मेरे ढोलना सुन मेरे ढोलना सुन मेरे प्यार की धुन मेरे ढोलना सुन मेरी चाहते तो फिजा में बहेंगी जिंदा रहेंगी होके फना साथी रे मर के भी तुझको चाहेगा दिल तुझे ही बेचैनियों में आएगा दिल मेरे गीतों के साए में तेरी राहतों की खुशबू है तेरे बगैर क्या जीना मेरे रोम रोम में तू है मेरी चूड़ियों की खन खन से तेरी सदाएं आती है ये दूरियां हमेशा ही नजदीक हम बुलाती है ओ पिया
మగసని ధని సగమా మగసని ధని సగ మగసని ధని సగమ సని ద మగ మదని సని ద మగ మదని సాని ధని స ధని స ధని స ధని స మదని 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 ధని స ధని స ధని స Madani, Madani, Madani. Madanisa, 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 Madanisa. Sanidama, 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 Sanidama.
मेरे ढोलना सुन, मेरे प्यार की धुन, मेरे ढोलना।
It looks like you have enjoyed the performance very well. Thanks, Sushmita, for such a fabulous performance. Doing a PhD is a stimulating, educating, enduring, and courageous task. How researchers feel, what challenges they encounter during their PhD, and how it feels at the end of journey, our performers are going to show that in the form of a skit. So please welcome THSTI's theatre group to perform the play, which is called as PPOP. Yes, it's PPOP. Before that, I would like to make a disclaimer here. All the dialogues and the names used in the skit are not related to any live human being, and these are purely fictional, and the play is not inspired by true life events. So let's welcome our theatre group.
सुनो सुनो सुनो, क्या खबर सुनाने आई हो?
अरे नहीं नहीं, खबर नहीं, कहानी सुनाने आई हूँ।
हाँ, कहानी, 4 दोस्तों की कहानी, उनकी पीएचडी की कहानी। आज मैं श्रुति,
मैं साधना,
आपको सुनाएंगे 4 दोस्तों की कहानी, उनके जीवन का सार। आइए सुनाते हैं आपको इस बार।
कैसे ये 4 दोस्त अमन, भूमिका, चेतना और दीपक मिले पीएचडी के 15 साल बाद,
और कैसे यादों की हवा उड़ा के ले गई इन्हें समय के उस पार।
तो आइए आपको लेकर चलते हैं द बेस्ट रिसर्च इंस्टीट्यूट TBRI के एलुमिनाई मीट में, जहां अमन अपने खाने की प्लेट पर ध्यान दे रहा है।
अरे माना सब बड़े बड़े लोग हैं, लेकिन ध्यान दे भी क्यों ना। हर फंक्शन में खाना भी तो जरूरी होता है, क्यों?
खाना भी? अरे नहीं, खाना ही जरूरी होता है।
अरे यार, नान तो हो गए, लेकिन कढ़ाई पनीर कहाँ है? ये रहा कढ़ाई पनीर। कैसे बचे? अरे अब डेजर्ट की जगह तो रही नहीं।
करेगा अमन, अब तो सुधर जा।
चेतना तू?
हाँ मैं।
कैसी है?
मैं ठीक हूँ, तू बता कैसा है?
अरे यार, मैंने देखा फॉक्सफर्ड से आई है तू, है सीनियर साइंटिस्ट हो गई, ऐश है तेरी।
अरे इतनी भी बड़ी बात नहीं, सब सही चल रहा है। तू बता ना, तेरा क्या हाल है?
बस टेम्स में सीनियर साइंटिस्ट बन गया हूँ, और फंडिंग का तो तू जानती ही होगी।
अमन, यू आर राइट, ये इशू तो मुझे वहां भी होता है।
अच्छा, अरे वहां से याद आया, तू फॉक्सफोर्ड से सिर्फ एल्यूमनाई मीट के लिए आई है? सच बता, या कोई और कोलैबोरेशन है तेरा?
अमन यार प्लीज, 15 साल हो गए हैं, तुम लोगों की याद आ रही थी, मैं मिलने चली आई। एल्यूमनाई मीट तो सिर्फ बहाना था। तुझे याद है बबीता मैम क्या कहती थी? हर मौके का फायदा उठाना चाहिए।
बबीता मैम, यार बात तो सही है, थी बड़ी स्ट्रिक्ट सी, लेकिन बातें काम की सिखाई उन्होंने। हाँ, अरे उससे याद आया, हमारी बाकी की मंडली कहाँ है?
हाँ यार, अरे प्लेट से नजर हटेगी तभी तो बाकी की मंडली दिखेगी। भूमिका, चेतना, कहाँ थे तुम दोनों? चलो इस मैडम जी को तो मुझे पता है। अरे स्टार्टअप ही तो है, नथिंग टू ब्रैग अबाउट।
नथिंग टू ब्रैग अबाउट? करोड़ों का तो इसका टर्नओवर है। हाँ, और इतनी फंडिंग तो मेरी लैब में भी नहीं आती। नथिंग टू ब्रैग अबाउट।
अच्छा ठीक है, लेकिन दीपक जी,
आपकी तो मुझे अलग से क्लास लेनी पड़ेगी। ना स्वेटर, ना स्विंग दिन, और तो और मेरी शादी में भी नहीं आया, सिर्फ कॉफी मशीन भिजवा दी गिफ्ट में। वो अलग बात है, यूज तो मैं अभी भी करती हूँ।
हाँ हाँ, यूज तो कर रही है ना।
अच्छा अच्छा, वो सब ठीक है, लेकिन ये बताओ इतने सालों से तू था कहाँ मेरे भाई?
दोस्तों, गुस्सा जायज है तुम सबका, बट यू ऑल नो व्हाट हैपेंड बैक देन। याद है ना मेरी क्या प्रॉब्लम थी और मैंने पीएचडी क्यों छोड़ी?
यार मैंने ब्रोकर कर दिया।
ये लाइट्स
ये लाइट
मैंने झुमका चेतना बोल दिया।
तो आइए दोस्तों, देखते हैं कटप्पा ने बाहुबली को क्यों मारा। अरे फिल्मी कीड़े, बाहुबली नहीं TBRI की कहानी है। अच्छा पहले ये बताओ, ये चारों आखिर कैसे मिले, और ये जेठा जी और ये बबीता जी कौन है? आइए दिखाते हैं ये चारों आखिर कैसे मिले, कहाँ मिले, कब मिले। TBRI के इंटरव्यू में।
ये पीएचडी इंटरव्यू कहाँ चल रहा है? एक्सक्यूज मी, ये पीएचडी इंटरव्यू यहीं चल रहा है?
जी चल तो यहीं रहे हैं।
ओके।
अरे 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 सर, ये स्टूडेंट्स के पीएचडी इंटरव्यू हैं। और
हाँ हाँ मानता हूँ, एज में थोड़ा सा बड़ा दिखता हूँ, स्टूडेंट ही हूँ। भगवान की दुआ से लास्ट अटेम्प्ट में एग्जाम क्लियर हुआ है।
अरे यार इतना टाइम हो गया, सुबह से आई हुई हूँ, इंटरव्यू कब स्टार्ट होंगे? देख के लग नहीं रहा कि पैनलिस्ट अभी तक आए हैं।
आप जानती है पैनलिस्ट में कौन है?
जानती तो नहीं हूँ, बट सीन्स लाइक वर्क हियर तो पहचानती हूँ।
अच्छा अच्छा, प्रोजेक्ट में होंगी आप?
हाँ जी।
नहीं नहीं, अभी तो नहीं आ पाएंगे सिंस वी आर स्टक विद दीस पीएचडी इनटेक इंटरव्यूज। बट डोंट वरी, फूड काउंटर खुलने से पहले वील बी देयर। क्यों जेठा जी?
जी बबीता जी, वैसे भी पीएचडी वालों का कितनी अच्छी केटरिंग होती है उनकी हर साल।
एब्सोल्युटली राइट जेठा जी, बट पहले यहाँ से तो फारिग हो। रजनीकांत, रजनीकांत, डोंट माइंड जेठा जी, डोंट माइंड जेठा जी, आपका असिस्टेंट है बड़ा स्लो।
मैडम इंटरव्यू स्टार्ट करवा दीजिए, मौसम बहुत गर्म है, बच्चे पिघल ना जाएं।
रजनीकांत, इतना दिमाग बातें बनाने में लगा लेते हो, काम में भी लगा लिया करो। ऐसे कैसे खड़े, ठीक से खड़े।
जेठा जी आपको मैंने
बच्चों को बुला लो।
बताया था ना आपको, हेल्थ खराब होती है स्ट्रेस लेने से।
अरे अमन कौन है? जी सर मैं हूं अमन। तुम्हारा नंबर जाओ अंदर। जी सर, मैं कम इन?
प्लीज प्लीज आओ।
गुड मॉर्निंग सर। गुड मॉर्निंग मैम।
गुड मॉर्निंग।
बैठो बैठो, मैं चेयर ला देता हूँ।
कोई बात नहीं सर, आप तो सर बहुत बड़े हैं सर, ऐसी भी क्या बात हो गई सर। नहीं नहीं सर, मैं सर ऐसे ही दूंगा इंटरव्यू सर। बताइए सर।
अमन राइट?
जी सर, जी जी।
काफी, तुम्हारा जेआरएफ तो 2 साल पहले ही क्लियर हो गया था।
जी
मास्टर्स
जी
नहीं ना?
जी सर।
काफी इंटेलिजेंट लगते हो। अच्छा ये बताओ, एच पाइलोरी का फुल फॉर्म क्या होता है?
एच पाइलोरी, हेलीकॉप्टर पाइलोरी सर।
हेलीकॉप्टर, तो आप सुन लो।
हाँ हाँ, okay okay, calm down. That'll be fine, Aman. My next question to you is, where do you see yourself in the next 10 years?
नेक्स्ट 10 years मैम, मैं एक बहुत बड़ा साइंटिस्ट बनना चाहता हूँ, श्री M.K. भान जी की तरह।
इतना इजी नहीं होता, इट्स अ टफ जर्नी।
I think we are done with you. Aman, I think we are done with you. Thank you so much.
थैंक यू मैम, थैंक यू सर।
नेक्स्ट, भूमिका कौन है? जाओ अंदर।
जेठा जी और बबीता जी ने किए बच्चों पर प्रश्नों के वार। क्या इन चारों का सिलेक्शन हो पाएगा इस बार? माना हिचकिचाहट थी इन सभी के जवाबों में, मगर कुछ तो देखा होगा इंटरव्यूअर्स ने इन नवाबों में। कौन डूबेगा और किसकी होगी नैया पार? क्या टीपीआरआई पहुंच पाएंगे ये चार? हाँ हाँ, चुन लिया चारों को, मगर अब होंगे ये और परेशान, क्योंकि अब शुरू होगा आगे का घमासान।
यार दीपक सुनना यार।
सुन रहा हूँ।
वो देख यार सीनियर्स को क्या कूल कूल एक्सपेरिमेंट्स करते रहते हैं पूरे दिन। असली रिसर्च तो यही कर रहे हैं, हम तो बस यार।
भूमिका शांत रहे, एक तो पेपर की लैंग्वेज समझ नहीं आ रही।
सुन रहा है तू?
रो रहा हूं मैं।
इनको लग रहा है जैसे हमारी लाइफ कितनी वंडरफुल है।
जब जेठा डेटा मांगेगा ना तब पता चलेगा।
तब इन्हीं दिनों को याद करके तस्वीर भाएंगे ये लोग।
अरे वाह, बहुत अच्छे। यहाँ क्यों सो रहे हो? केबिन में जाओ, वहां एसी स्ट्रांग है, वहां की चेयर्स भी कंफर्टेबल है। लेजी फेलो। और हाँ, क्या चल रहा है तुम लोगों का?
वो पीसीआर में जो पेन आना था वो तो नहीं आया, जो नहीं आना था वो आ गया।
वाह वाह, बहुत अच्छे। और तुमने क्या कहा था? ये डेटा पैनसेट में जाएगा। ऐसे ही मेरे लैब का रिएजेंट वेस्ट करना है तो कल से लैब मत आना, स्टूपिड फेलोज। और तुम लोगों को जो रिव्यू लिखने का काम दिया था, क्या हुआ?
सर आई सेंट यू द ड्राफ्ट, यू कैन चेक।
ओके सर। और दीपक आपका?
सर सच कहूँ तो मैं कोशिश कर रहा हूँ, लेकिन ये पेपर की लैंग्वेज समझ नहीं आ रही। सर आपके पास अगर टाइम हो तो मुझे समझा सकते हो, मैं रिव्यू पेपर लिखने का शुरुआत करूँ तो करूँ?
अरे दत्त, किसी को इस लैब में सोना है, किसी का डेटा नहीं आ रहा है, किसी को पेपर पढ़ना नहीं आ रहा है। ऐसे चलेगी मेरी लैब? ऐसे आएगी फंडिंग? हेलो, हाँ कौन?
जेठा जी।
बबीता जी।
काफी गुस्से में मालूम होते हैं, सब ठीक तो है?
अरे नहीं नहीं बबीता जी, आपसे कैसे नाराज हो सकते हैं? ये मेरी लैब है ना।
जेठा जी, आई हैव टोल्ड यू 1000 टाइम्स नाउ, स्ट्रेस लेने से हेल्थ खराब होती है। एंड प्लस बच्चे ही तो हैं, विल लर्न। और वैसे भी आज तो आपको खुश होना चाहिए, फाइनली सीबीटी का प्रोजेक्ट अप्रूव हो गया। सो वेयर इज द पार्टी?
अरे बबीता जी, पार्टी तो आपकी बनती है, मेरा तो सीएसटी का प्रोजेक्ट है, आपका तो इंटरनेशनल बेट्स फाउंडेशन।
या दैट्स राइट, बट जेठा जी मानना पड़ेगा, पैसे बचाने का एक भी मौका छोड़ते नहीं है आप। और एज यू नो, प्रोजेक्ट अप्रूव तो हो जाता है, फंड्स तब आते हैं जब फर्स्ट रिपोर्ट सबमिट करने की डेडलाइन आ जाती है। ओ, डेडलाइन से याद आया, मेरी नेक्स्ट मीटिंग का टाइम हो गया, आई विल कैच अप विद यू लेटर।
लेकिन बबीता जी चाय पे मिले। हेलो हेलो बबीता जी।
लगता है मैडम फोन काट दी, आपकी पेन।
अरे जाके काम करो अपना, लेजी घड़ी का।
अमन, अमन।
यस मैम, आपने बुलाया?
इतना टाइम लगता है वहां से आने में? Where is the update? Like 4 days ago, I think I asked you for an update. Where is it?
4 days ago update?
अरे बेटा, सीबीटी के प्रोजेक्ट के फंड्स की डिटेल्स मांगी थी मैंने फाइनेंस से। Where are they?
हाँ जी हाँ जी हाँ जी मैम, आई वॉज बिजी विद माई एक्सपेरिमेंट्स, तो मैं भूल गया।
अमन, 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 एक काम करती हूँ, मैं भी भूल जाती हूँ that you are my PhD student. Why don't you realize how important this is for me? Tomorrow, the first thing I want is this on my table. And go out now and
Okay, ma'am.
send Chetna in while you're out.
ओके मैम। चेतना,
हाँ बोल।
सुनना।
हाँ बोल ना।
तुमने कुछ किया है क्या?
नहीं तो।
यार मैडम ना थोड़े गुस्से में है।
अरे क्या क्या हुआ यार, डरा क्यों रहा है?
तुझे बुला रही है और ना बस चुपचाप सुन के आ जाइयो।
तुझे डांट पड़ी?
ना 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 ना, ना मुझे कुछ नहीं कहा। मैं तो फेवरेट स्टूडेंट हूँ, लेकिन संभाल लियो, ठीक है?
मैम मे आई कम इन? आपने बुलाया?
I went through your manuscript. It was fine. I've made some minor edits. Just check it once.
Okay, ma'am, I will.
Lab में सब ठीक चल रहा है?
Yes, ma'am.
Okay. These admin people have sent me a mail, Foundation Day ke regarding, and I want you to coordinate it.
No, ma'am, but I can't, ma'am. Sorry.
बच्चे, such events prepare you to become a global leader. And take it from me, personal and professional, दोनों life में काम आएगी.
Okay, ma'am, I will do.
Now I'm leaving for my next meeting. Talk to me later.
Sure, ma'am.
तो देखा आप लोगों ने, वहां बेचारा दीपक कर रहा था पेपर पढ़ने का स्ट्रगल और सोच रहा था क्या पीएचडी में सबको होता है ये ट्रबल। भूमिका और चेतना की सही चल रही थी गाड़ी, मगर अमन पर कल पड़ेंगे एडमिन और फाइनेंस के सवाल भारी। अच्छा, अब ये बताओ कहानी में हुआ क्या आगे? मैं क्यों बताऊं, तुम खुद ही देख लो आगे। तो चलो।
एडमिन सर एडमिन यही है क्या?
मुबारक हो, एडमिन में स्वागत है आपका। नमस्ते नमस्ते। अरे बॉस तो खुद ही सीबीटी के ऑफिस गए हैं आज, वो मीटिंग थी ना उनकी। एक सेकंड खड़े हैं। वैसे बच गई आप उषा जी, बुरा ना मानना, पंचालिटी इज अ की टू सक्सेस। हाँ अरे उषा जी, समोसे नहीं, थोड़ा हेल्थ कॉन्शियस हो गया हूं ना आजकल। वो मिसेस ने ना डाइट पे रखा हुआ है, आप तो जानती है मिसेस को। चाय ले लूंगा बट दैट टू विदाउट शुगर, विदाउट मिल्क। हाँ, तुम बताओ अब।
सर इंक्वायरी थी, बबीता मैम ने भेजा था।
अरे बबीता जी के साथ क्या आए हो, पहले बताना था ना। तो बताओ क्या चलना है?
सर बबीता मैम का जो प्रोजेक्ट है सीबीटी के नाम का। सर
सीबीटी? बेटा लगता है नए आए हो, सीबीटी नहीं एबीटी होता है। तो बताओ क्या जानना है?
सर वो प्रोजेक्ट के बारे में जानना था।
प्रोजेक्ट के बारे में? प्रोजेक्ट का नंबर तो दे दो बेटा।
नंबर तो नहीं पता सर।
प्रोजेक्ट का नाम
बड़ा सा था, कुछ ड्रॉट रेजिस्टेंस के जैसा था कुछ।
बेटा ड्रॉट रेजिस्टेंस पे तो पूरा टीवीआरआई काम कर रहा है, ऐसे कैसे बताऊंगा? मैडम ने फाइल तो दी होगी।
फाइल भी नहीं दी सर देख लीजिए ना सर, अभी डांट खा के आया हूँ सर, देख लीजिए सर प्लीज।
बेटा प्रोजेक्ट का नाम नहीं पता, नंबर नहीं पता, कहाँ से निकालूं फाइल?
देख लीजिए सर प्लीज। सर प्लीज
रुको, नए आए हो इसलिए तुम्हारी हेल्प कर रहा हूँ। शर्मा जी सर। एक काम करो, बबीता जी के जितने प्रोजेक्ट हैं सभी फाइलें ले आओ।
सभी फाइलें?
हाँ मिस्टर शर्मा, सभी फाइलें। अच्छा, अगली बार से फाइल आनी चाहिए जिस प्रोजेक्ट के बारे में जानने आए हो।
जी सर।
अरे शुक्ला, एक काम करो, उषा जी कुछ समोसों के लिए कह रही थी यार। हाँ, तो ये फाइल, हाँ ये रही फाइल। तो बताओ क्या जानना था?
सर इसके पूछने थे फंड्स कब आएंगे सर?
फंड? फंड?
जी
बेटा ये कौन सा डिपार्टमेंट है?
सर एडमिन।
एडमिन। बेटा अगर फंड्स के बारे में जानना था तो फाइनेंस सेक्शन जाना था ना। अब ये भी काम भी एडमिन बताएगा क्या? वो देखो वित्त विभाग की उषा जी बैठी हैं, उनसे जाके पूछो।
ठीक है सर, ठीक है सर।
अरे बेटा फाइल तो लेके जाओ। क्या पूछोगे वहां जाके?
जी सर, सॉरी सर। मैम।
प्रेशर हो?
जी
यही से डांट खा के आए हो?
जी जी
अरे
मैम।
कोई बात नहीं बेटा, अभी तो 5 साल बाकी है, अभी बहुत आगे जाना है, ऐसे परेशान नहीं होते। लाओ दिखाओ क्या लाए हो।
मैम ये शुक्ला जी नहीं है?
हाँ हाँ।
उन्होंने फाइल भिजवाई थी, बबीता मैम का प्रोजेक्ट है।
अच्छा क्या जानना है इसमें?
मैम इसके फंड्स कब आएंगे?
कौन सा प्रोजेक्ट है ये?
ड्रॉट रेजिस्टेंस।
ओह ड्रॉट नहीं, रुको लेट मी चेक। अरे ये तो अभी 2 महीने पहले सैंक्शन हुआ है, इसके फंड्स आने में अभी कम से कम 6 महीने लगेंगे।
6 महीने?
हाँ।
तो बबीता मैडम को क्या बोलूं?
यही बोलना कि जब भी फंड्स आएंगे मैं उन्हें खुद बता दूंगी, टेंशन ना ले ज्यादा, मैं खुद फोन कर दूंगी।
खुद बता देंगी?
हाँ मैं खुद बता
हाँ
दूंगी।
बच गया।
और कुछ?
थैंक यू। नहीं मैम बढ़िया, आप बताइए।
अरे काम में और कुछ?
नहीं मैम बस बढ़िया, बस ठीक है।
जाओ फिर।
थैंक यू मैम।
शर्मा जी। हाँ शुक्ला जी, गर्मा गर्म चाय समोसे आ गए हैं, पर आप तो सिर्फ चाय ही लेंगे ना?
अरे उषा जी सब गरम गरम समोसे आ ही गए हैं तो एक दो तो हम भी ले ही लेंगे।
आइए आइए, समोसे के मजे लिए जाए।
तो दोस्तों देखी आपने इनकी रोज रोज की कहानी। आगे क्या होता है सुनिए मेरी जुबानी। गाड़ी चल तो पड़ी थी इनकी पीएचडी की, मगर रास्ते में स्पीड ब्रेकर्स कहाँ कम है। एक्सपेरिमेंट्स, स्टाइपेंड्स और जर्नल्स की दुनिया में ये सारे गुम हैं। लेकिन ये दीपक को क्या हुआ है? इसे किस बात का गम है? क्या मेस के खाने में मसाला कम है? नहीं नहीं, बात कुछ और ही है। आइए देखते हैं। तो चलिए।
यार भूमिका, यार आज संडे मूवी देखने चले क्या? बता ना यार।
यार मूवी पे क्या पैसे वेस्ट करने हैं, इतनी सी तो स्टाइपेंड है।
अरे यार
बाहर ना मोमोज वाले की नई दुकान खुली है, वहां मोमोज खाने चलते हैं।
नहीं यार।
अरे यार आज फिर से रोटी में बाल। क्या है ये सब? तंग आ गया हूं मैं इस खाने से।
यार बाल ही तो है, प्रोटीन ही तो है। लुक एट द ब्राइट साइड यार। ऊपर से तेरे को पता है क्या?
क्या?
परसों ना दीपक के पोहे में बीड़ी निकली थी।
लेकिन ये तो है कि केराटिन इज स्टिल बेटर देन कार्सिनोजन। क्यों दीपक?
अरे रुको रुको दीपक, तेरा मुंह इतना लटका हुआ क्यों है आज? क्या हुआ?
हाँ मुझे भी आज हमारा दीपक थोड़ा बुझा बुझा लग रहा है।
हाँ यार, कहीं उससे बात नहीं हुई इसलिए तो बुझा बुझा तो नहीं है?
वो कौन है?
अरे वो
वनराज सर की स्टूडेंट। क्या
नाम है उसका?
दीपिका।
अरे नहीं यार, हर वक्त वही मजाक।
नहीं नहीं दोस्तों रुको, बात कुछ और है। दीपक घर पे सब ठीक है?
हाँ दोस्तों घर पे सब ठीक है।
तो हुआ क्या?
जब से पीएचडी में एडमिशन हुआ है, जेठा सर ने एक ही काम दिया है, रिव्यू पेपर लिखना, और वो भी मेरे से हो नहीं रहा। और सर भी क्या कर रहे हैं, हर पेपर अलग, हर पेपर के लैंग्वेज अलग, बहुत कॉम्प्लिकेटेड है। अच्छा बताओ तुम लोग पेपर कैसे लिखते हो?
अरे
यार दीपक तू बैठ, मैं तुझे बताती हूँ। तुझे रिव्यू पेपर लिखना नहीं आ रहा, मैं तुझे बताती हूँ हाउ टू राइट पेपर इन 5 days।
अच्छा वो कैसे?
देख पहले ना तुझे अपना मिलता जुलता पेपर ढूंढना है, वो तेरा क्या बन जाएगा? टेम्पलेट। फिर क्या? लैंग्वेज चेंज, कॉपी पेस्ट। उसके बाद क्या? तुझे अपना फॉन्ट क्या करना है? टाइम्स न्यू रोमन, साइजिंग 12 स्पेस 1.5। बस यार रेफरेंसेस डालने मत भूल जाइयो।
अरे हाँ फ्लैग भी चेक कर लियो।
हाँ
लैंग्वेज चेंज, कॉपी पेस्ट। दोस्तों साइंस करने आया हूं यहाँ, इस पेपर के पीछे की साइंस समझे बिना इस कॉपी पेस्ट का क्या फायदा? तुम सब बड़े बड़े जगहों से पढ़ के आए हो, डेजर्टेशन करके आए हो, मुझे लगा तुम्हें पता होगा कि साइंटिफिक राइटिंग क्या होती है, डेटा इंटरप्रिटेशन क्या होता है।
तुझे क्या लगता है डेजर्टेशन में क्या साइंटिफिक राइटिंग लिखना सिखाते हैं? अबे मीडिया बनवाते हैं, बफर बनवाते हैं। और तो और टीनियर्स अपने बर्तन धुलवाते हैं यार। अबे बर्तन नहीं, बीकर।
बीकर धुलवाते हैं।
हाँ हाँ बीकर।
ये गलत है। मैं साइंस से प्यार करता हूँ, इसलिए मैंने पीएचडी चुनी। और ये तुम सब जो बता रहे हो, ये काम चलाने का तरीका है।
अरे थोड़े समय की तो बात है, काम चलाओ तरीके से ही सही, नाम के आगे डॉक्टर तो लग जाएगा। जस्ट इमेजिन, डॉक्टर चेतना सुहानी। अरे वाह, डॉक्टर चेतना सुहानी।
चेतना, मुझे टाइटल नहीं चाहिए। इस टाइटल का क्या फायदा जिसका कोई फायदा उठा ना सकूं मैं।
अरे तू बैठ, सुन। अब हर पीआई अगर पेपर समझाता रहा और स्पून फीड करता रहा तो कैसे काम चलेगा। लेकिन एक बात तो है, अगर ये सब चीजें हमें मास्टर्स में सिखाई जाए तो सही ना रहे।
हाँ यार, जैसे मास्टर्स में साइंस के फंडामेंटल सिखाए जाते थे, ऐसे ही साइंटिफिक राइटिंग का भी कोई सब्जेक्ट तो होना चाहिए, है ना?
इनफैक्ट पता है क्या, साइंटिफिक लैंग्वेज क्या है, इंटरप्रिटेशन, इसके अलावा प्रपोजल कैसे लिखें, फंडिंग एजेंसी कौन सी होती है, नेशनल हो इंटरनेशनल हो, ये सब भी तो बताया जाना चाहिए। क्यों?
हाँ यार।
मैं तब से तुम लोग को यही समझा रहा हूँ, साइंस समझना ही नहीं, साइंस को सही तरीके से करना आना भी चाहिए, तभी तो साइंस करने में मजा आएगा।
अरे 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 अरे, बंद क्यों कर दी ये कहानी? अभी तो समझ आने लगी थी पीएचडी स्टूडेंट्स की परेशानी। अब स्टोरी में आगे क्या हुआ ये हमें कौन बताएगा? क्या इन चारों को साइंस करने का मजा भी आएगा? अरे दुखी मत हो, अभी तो कहानी है काफी बाकी। अभी है सीजीसी के डायरेक्टर डॉक्टर जेठा की एंट्री बाकी। डॉक्टर जेठा डायरेक्टर बन गए? हाँ। ये कब हुआ और हम लोग ही कहाँ आ गए? अरे अभी तक तो चल रही थी ये कहानी भूतकाल में, इन्हें वापस भी तो लाना है वर्तमान में। ओ वहीं जहाँ टीबीआरआई का एलुमनाई मीट चल रहा है। हाँ। और ये चारों दोस्त मिलते हैं पीएचडी के 15 साल बाद, और जहाँ इन सब की नजरें ढूंढ रही थी किसी व्यक्ति विशेष को, और ढूंढे भी क्यों ना, ढूंढने की वजह भी कही थी।
दोस्तों, गुस्सा जायज है तुम सबका, बट यू ऑल नो व्हाट हैपेंड बैक देन। याद है ना मेरी प्रॉब्लम क्या थी और मैंने पीएचडी क्यों छोड़ी? वो हम लोग रिसर्च में क्या कहते हैं, ट्रबल शूटिंग कर रहा था इन दी लैब ऑफ लाइफ। अच्छा सुनो, जेठा सर और बबीता मैम कहाँ है?
हाँ यार, बबीता मैम दिखाई नहीं दे रही, वो तो बिजी होंगी ना, प्रिंसिपल साइंटिफिक एडवाइजर जो बन गई है। और जेठा सर ना सीजीसी के डायरेक्टर बन गए, डायरेक्टर।
अरे सीजीसी नहीं, सीजीएसटी के डायरेक्टर हैं।
हाँ यार, सीजीएसटी जो भी है, आज के चीफ गेस्ट भी वही है।
वो
आते ही होंगे।
अब आते हैं तो मिलेंगे।
ठीक है
लेकिन आ रहे हैं तो मैं अपनी फंडिंग की बात भी कर लूंगा।
मैं ना अपने सायरे के प्रपोजल के बारे में बात कर लूंगी।
काम भी कर लेते।
अरे एलुमनाई मीट में भी काम की बातें। चिल करो, काम तो होता रहेगा। देखो तुम्हारी बबीता मैम भी तो आई है।
हेलो सर।
हेलो मैम। हेलो। और आती भी क्यों ना, बच्चों से मिलने का इससे अच्छा मौका कभी मिल सकता है। बाय द वे, वी आर एब्सोल्युटली इम्प्रेस्ड विथ योर सक्सेस।
स्पेशली यू दीपक, कैसे हो?
ठीक हूँ सर।
मैंने तुम्हारा नया रिफॉर्म देखा, पीपीओपी रिफॉर्म। इट्स रियली गुड। आई एम रियली इम्प्रेस्ड विथ योर सक्सेस।
थैंक यू सर।
बट आई फेल्ट रियली बैड जब तुम लैब छोड़ के चले गए। आई वॉज वेरी मच सैड, बट था तुम में पोटेंशियल, बट शायद हमसे ही कुछ।
नहीं सर, मेरा अल्टीमेट गोल तो साइंस में कंट्रीब्यूट करना था, चाहे अपने पीएचडी के थ्रू या दूसरों के पीएचडी आसान करने की तरफ हो।
अच्छा, इसीलिए तुमने ये पीपीओपी रिफॉर्म पास करवाया है?
जी सर।
वेरी गुड, वेरी गुड।
ये पीपीओपी रिफॉर्म क्या है?
आप सबको पीपीओपी रिफॉर्म के बारे में नहीं पता? पीपीओपी रिफॉर्म है प्री पीएचडी ओरिएंटेशन प्रोग्राम। इस रिफॉर्म के जरिए मास्टर्स और पीएचडी के स्किल सेट गैप को कवर किया जाएगा। मास्टर्स में ही बच्चों को दी जाएगी पेपर पढ़ने की सही जानकारी और उसको कैसे इंटरप्रेट करें वो भी सिखाया जाएगा। इसके साथ साथ सिखाया जाएगा कि कौन कौन सी नेशनल इंटरनेशनल फंडिंग एजेंसीज अवेलेबल है और उन फंडिंग एजेंसीज को कैसे अप्रोच करें, कैसे प्रपोजल लिखें। ये रिफॉर्म हमारे अपने दीपक की तो देन है, क्यों जेठा जी?
जी बबीता जी, तो देखा आप लोगों ने, बिना पीएचडी के भी आप साइंस में कंट्रीब्यूट कर सकते हैं। पर्दे के पीछे रहकर भी साइंस को बेहतर से बेहतर बनाया जा सकता है। तो आइए आज से ये ठान लेते हैं कि किसी भी विद्यार्थी को कम नहीं आंकेंगे, क्योंकि हर एक विद्यार्थी किसी ना किसी तरीके से समाज और विज्ञान को और ऊपर ले जा सकता है। सो हमारे दीपक ने ये प्रूव कर दिया कि where there is a will, there is a way.
Absolutely right, जेठा जी।
कल तक किसी की लैब में थी फंड्स की किल्लत, तो किसी को हो रही थी पेपर लिखने में दिक्कत। पीपीओपी में मास्टर्स में ही दी गई फंडिंग एजेंसीज की जानकारी और साथ में दूर की गई स्टूडेंट्स के राइटर्स ब्लॉक की बीमारी। दीपक के रिफॉर्म से ना सिर्फ खुश हुए पीआई सारे, बल्कि स्टूडेंट्स के भी हो गए वारे न्यारे। कहाँ हमें लग रहा था दीपक ने कर दिया एक सीट बर्बाद, हमें क्या पता था दीपक करेगा हजारों जिंदगियां आबाद।
हम होंगे कामयाब, हम होंगे कामयाब, हम होंगे कामयाब एक दिन। ओ मन में है विश्वास, पूरा है विश्वास, हम होंगे कामयाब एक दिन।
सब होंगे
कामयाब एक दिन।
आशा करता हूं आपको हम सबकी प्रस्तुति पसंद आई होगी, तो क्यों ना हम अपने कलाकारों से रूबरू हो जाएं।
I'm साधना as narrator,
मेरा नाम अभिषेक है, प्लेइंग एज शुक्ला जी।
पूजा as चेतना, समृद्धि as भूमिका,
स्नेह as डॉक्टर बबीता,
सौविक as डॉक्टर जेठा,
सोन as दीपक,
रोहित as अमन,
मोनिका as उषा जी,
अखिलेश एज रजनीकांत, साहिल एज सीनियर एंड शर्मा जी,
ममता as senior, श्रुति as narrator,
सोमोदीप एज लाइट डायरेक्शन।
Ma'am is fictitious. The characters are fictitious.
Yes.
Inspired by real life events.
No, not inspired. No, we are not inspired by any real life event.
Thank you so much. Thank you.
Thanks very much. That was a wonderful performance by our group. I would like to mention here special thanks to Shomodeep for lights, Sneh Bajpayee for script, Rohit Kachwaha for direction, and special thanks goes to Mr. Sudhir Kumar for playing a major role as a mentor during the refinement of this play.
Sneh, can we please have you on the stage? If we have Sudhir sir here.
वो अच्छा था, चेयर नहीं लगा दूं। इन्होंने अच्छा कितना चेयर नहीं लगा दूं।
Sudhir sir.
आप लोगों को अच्छा लगा होगा, क्योंकि टाइम बहुत कम था और टारगेट बड़ा था, बट उम्मीद करता हूँ बच्चों को बहुत कम टाइम भी मिला और हम कोऑर्डिनेट भी ढंग से कर नहीं पाए। और आप लोगों को तालियां बता रही थी कि एफर्ट्स वाज इन राइट डायरेक्शन। मेरी आवाज बहुत है। तो एफर्ट्स वाज इन राइट डायरेक्शन, क्योंकि आप लोगों ने जितना एंजॉय किया, और उम्मीद करता हूं कि रंगमंच को सिर्फ कुछ चीजें आज तक जिंदा रख रही हैं, उसमें कॉलेज और यूनिवर्सिटीज का बहुत बड़ा योगदान है कि रंगमंच आज तक जिंदा है। मैं आप सभी से रिक्वेस्ट करूंगा कि रंगमंच को इस तरह जिंदा रखें और ये एक्टिविटीज बच्चों को ना सिर्फ प्रेरित करती हैं आगे बढ़ने के लिए, बल्कि इनको जिंदगी के बाकी पहलुओं के बारे में भी बहुत जानकारी देती हैं। तो मैं आप सभी से अनुरोध करता हूं कि रंगमंच को यूं ही आगे बढ़ाते रहेंगे। बहुत बहुत शुक्रिया। थैंक यू वेरी मच अपना समय देने के लिए। थैंक यू, थैंक यू,
थैंक यू सुधीर जी। Music. It fills our soul with serenity, calmness, and evokes spiritual experience. The art is popular not as a form of entertainment, but also as a means of connecting with divine being. And being a neuroscientist, I must add that just like exercise, music has been shown to increase the levels of oxytocin and serotonin in your brain. And both of these hormones are instant mood boosters. So here we are presenting our researcher-singer combos whose soothing voice and melodious performance will stir your heart. So let's put your hands together for our first singer, Abhishek.
Hello.
करिया मेरिया भूल गया हर की करना जीतनू लगाती तो कांपती थी पलके मायरी चुनिया सजा के वो देती वादे कल के मायरी मेरे हाथों खौफ का थी चाशनी हर की बात मायरी याद वो जन्नता लंगरी आयरी मन्नता मंगरी आयरी याद वो बारिशों में फटके माँ आती थी वो चल के मायरी तेरिया हो जाए तो रोती हल्के हल्के मायरी फिर से मैं रोऊं फिर वो गाए ठंडी हवाएं बन के छाए मायरी गीत वो पानी मायरी हीरा वो गांधी मायरी याद वो आएगी मायरी से कहूँ मायरी अब क्या करूँ से कहूँ
थैंक यू।
So I would like to invite our next singer, Shweta.
सोचा कहाँ था ये जो हो गया, माना कहाँ था ये हो गया, कदमों को थामो ना है हम तो होश में, कदमों को थामो ये है उड़ते जोश में, बादल पे कहाँ है या झूठा क्या है, अब तो उड़ कर पड़ी अपनी ये नाव है, बादल पे कहाँ है या झूठा क्या है, अब तो उड़ कर पड़ी अपनी ये नाव है। चल पड़ी सफर अजनबी तो हट गए, लगता हमको मगर कुछ कर देंगे हम मगर, ख्वाब में जो लिखा वो था छिपा फंस जाएगा भूल गए, रे 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 बादल पे कहाँ है या झूठा क्या है, अब तोड़ चल पड़ी अपनी ये नाव है, बादल पे कहाँ है या झूठा गाँव है, अब तोड़ चल पड़ी अपनी ये नाव है, ये ओ,
Thanks, Shweta, for such a nice performance. I'm sure that their singing is soothing your souls. To bring more variety to your entertainment, we are introducing another category of musical instruments. Again, going by science, playing a musical instrument can enhance verbal memory, spatial reasoning, and it relieves stress. So I would like to call on stage Mr. Prashant, who will play Indian slide guitar.
ऑन नहीं है।
Good afternoon, everybody. Myself, Prashant Gulbal. I will be performing Indian slide guitar. There are 3 minutes given to me to just before going to give few more introduction, I will just say this is a song from film Mehbooba, composed by I.D. Burman. This song has been composed in Rag Shivranjani, one of the very favorites of the musical composers. खास करके शंकर जयकिशन का बहुत चहेता राग था है। जाने कहाँ गए वो लोग, बहारो फूल बरसाओ, दोस्त दोस्त न रहा, और अभी ये मेरा गीत।
Hello. So here we present Rohit, who will recite a piece composed on a classical rag, accompanied by Abhishek on the instrument.
ये लगेगा, ये लगेगा, ये लगेगा, ये लगेगा। ऐसे ऐसे रखना पड़ेगा ये। ठीक है, टेप ले लो। टेप है हमारे पास, टेप।
Check, 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 check. नमस्ते सभी को। इसका गेन बढ़ा दीजिए प्लीज। साउंड चल चल। मॉनिटर से आवाज चली गई सर। नमस्ते, मैं रोहित, मेरे साथ हैं अभिषेक जी। हम आपके समक्ष एक फ्यूजन कॉम्पोजिशन प्रस्तुत करेंगे जो राग खमाज और राग काफी में है। ये एक क्लासिकल फ्यूजन है जिसको साथ में राग जोग के नाम से कहा जाता है, क्योंकि राग जोग है तो योग से आया है ये शब्द, और बहुत ही सुंदर रस है, श्रृंगार रस में कहते हैं। बहुत रोमांटिक और बड़ी सटल सी इसके अंदर कॉम्पोजिशन बनाई जाती है। जो मैं एक बंदिश गाने वाला हूँ, उसके बोल कुछ ऐसे हैं। का से कहूँ सजन अपने मन की बतिया, बिन तुमरे जागी मैं सगरी रतिया। सो I mean, this is a story of everyone, be it work or you know. So yeah.
Sa गमा पम पनी पनी सा सन युगमा पम गा सा गा कैसे कहूँ सजन कैसे कहूँ सजन कैसे कहूँ अपने मन की बदिया कैसे कहूँ सजन कैसे कहूँ सजन कैसे कहूँ सगा 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 हा मगा 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 हा निसा 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 हा सानी बानी गा बनी सानी बानी गा मा गा मा गा बनी सा सानी पा मा मा बिन तुमरे जागी बिन तुमरे जागी जागी मैं जागी जागी मैं जागी बिन तुमरे जागी मैं बिन तुमरे जागी मैं सगनी रतिया सगनी रतिया सगनी रतिया बबली मग सगनी रतिया सगनी रतिया कैसे कहूँ कैसे कहूँ गम 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 सारी सारी परी गम 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 कैसे कहूँ कैसे कहूँ अपने मन की बदिया सजन 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 कैसे कहूँ थैंक यू बहुत बहुत धन्यवाद
Thanks Rohit and Abhishek for this melodious performance. Our next solo singer is Shavik, who will play acoustic guitar to reach your heart.
हेलो माइक ऑन नहीं है ऑन हेलो ऑन है
नंबर खड़ी
So I'm going to play two songs today. The first one is called Tu Nazm Nazm Sa by Orko Mukherjee, and it goes like Tu nazm nazm sa mere होठों पे ठहर जा मैं ख्वाब 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 सा तेरी आंखों में जागू रे, तू इश्क इश्क सा मेरे रूह में आके बस जा, इस ओर तेरी शहनाई उस ओर मैं भागू रे। हाथ थाम ले पिया करते हैं वादा, अब से तू आरज़ू तू ही है इरादा, मेरा नाम ले पिया मैं तेरी रुबाई, तेरे ही तो पीछे पीछे रात आई, सांसों में बिखर जा मैं फकीर तेरी गुर्बत का, तुझसे तू the departed soul, my friend and colleague Manjit Kumar. हो जाते नहीं कहीं रिश्ते पुराने, किसी नए के आ जाने से, जाता हूँ तो मुझे तू जाने दे, क्यों परेशान है मेरे जाने से, टूटा है तो जुड़ा है क्यों मेरी तरफ तू मुड़ा है क्यों, हक नहीं तू ये कहे कि यार अब हम ना रहे, मीठी सी वो गाली तेरी सुनने को तैयार हूँ। Thank you.
Thanks, Shovik, for this delightful performance. Our next solo singer is Shruti, who is here to pay a homage to musical gems our Indian music industry has lost in the last year. So over to you, Shruti.
Good evening, everyone. So, COVID has taken a lot from all of us, and it had a huge impact on the music industry too. So, whatever I do will never be enough to pay homage to them. It's just a small dedication to some of my favorites. हमको मिली है आज ये घड़ियां नसीब से, जी भर के देख लीजिए हमको करीब से, फिर आपके नसीब में ये रात हो न हो, शायद फिर इस जन्म में मुलाकात हो न हो, लग जा गले से है। सुरमई अखियों में नन्हा मुन्ना एक सपना दे जा रे,
निंदिया के उड़ते पाखी रे अखियों में आ जा साथी रे निंदिया के उड़ते पाखी रे अखियों में आ जा साथी रे रारे रारम ओ रारे रू रारे रारम ओ रारे रू चुपके से कहीं धीमे पाँव से जाने किस तरह किस घड़ी आगे बढ़ गए हमसे राह में पर तुम तो अभी थे यही कुछ भी ना कहा कब का था गिला कैसे कह दिया अलविदा but then there was somebody who said चलते चलते मेरे ये गीत याद रखना कभी अलविदा ना कहना कभी अलविदा ना कहना हंसते रोते बस यूं ही तुम गुनगुनाते रहना कभी अलविदा ना कहना कभी अलविदा ना कहना That's all from my side. Thank you.
Thanks, Shruti, for such a marvelous marvelous performance. Priya's voice will make feel afresh. Preeti.
अगलावा मजबूरी नु आड़ जाड़ दी पसूड़ी नु जह बने हाँ तेरी ये जावा मैं पूरी नु आड़ा सी आया दिल बाग बाग मेरा टकराया आगंदी नु रावा चुबावा चोरो लुका कोई में मेरे ढोल जुदाई आजा वो दिल तेरा पूरा भी ना हो बढ़िया बनाया भी बात की वो हो गए आजा वो दिल तेरा पूरा भी ना हो भूल गई मजबूरी नो दुनिया दी दस्तूरी नो साथ तेरा है पूरा जना दिखलाया दिल हमारा बेसहारा ख्वाहिशात अधूरी नो वारी मजावा में तनो बुला मगल सारी ता होवे मेरे ढोल जुदाई आदि तने खबर की वो होवे आ जावे दिल तेरा पूरा भी ना होवे हाँ बढ़िया बनाई आदि कल बात की वो होवे आ जावे दिल तेरा पूरा भी ना होवे मेरे ढोल जुदाई आदि मेरे ढोल जुदाइया दी सरदारी ना हो गए दिलदारा दी सब यारा दी आजारी ना हो गए सब यारा भी आजाद ना हो। आज चले लेके तुझे है जहां सिलसिले, तेरी कमी बना दे सजा दे पनाह दे हमें, बना दे सजा दे पनाह दे हमें। थैंक यू।
Thank you, Preeti, for such a stunning performance. Let's fill this Audi with some more energy. And let's put your feet to dance. Since our performers are going to perform the traditional and the most popular folk dance of Punjab. So any guess? Yes, you guessed it right. It's Bhangra. This dance form is extremely energetic and full of life. It's the newest dance, music, and fitness phenomena which is spreading all over the world. So here at THSTI, we just don't share the research ideas, but we also share the cultures from different states. So people from different states are going to perform Bhangra at this stage. So let's welcome Akhilesh, Sahil, Mamta, Kamli Priya, and Sadhna, who will energize the floor with the Bhangra. Thanks, Bhangra Group. Thanks, Bhangra Group, for such a powerful performance. Our next performer is multi-talented. I'm sure with her poem and dance, she's gonna rock the stage. So let's welcome Zohra.
Hello. Good evening, everyone. This is Zohra Hashmi. I am first year PhD student at THSTI, and today, on the occasion of Foundation Day, I want to try to strengthen this foundation with a small poem. This poem is for one and all THSTI family members, and a special dedication to my PhD colleagues and seniors. So the title of the poem is टूटना नहीं है। जिंदगी है, हर तरह के मोड़ मिलेंगे, इसके सफर में हर तरह के लोग मिलेंगे। हाँ, मगर विश्वास करने से ज्यादा विश्वासघात करने वाले मिलेंगे। ये कहने वाले मिलेंगे कि तेरे जीने का कोई वजूद नहीं है, और तुझे चलते रहना है कि तेरे हिम्मत का कोई तोड़ नहीं है। मुश्किलें तो बहुत आएंगी जिंदगी में, कभी आगे से तो कभी पीछे से आएंगी, बार बार और हर बार आएंगी। लड़ते लड़ते कभी थक जाओ तो थोड़ी देर ठहर जाना, पर इन मुसीबतों के आगे तुझे झुकना नहीं है। तेरी हिम्मत ही तेरी ताकत है, तुझे टूटना नहीं है। प्यार के नाम पे धोखे मिलेंगे, काम निकलवाने वाले दोस्त मिलेंगे। वक्त ऐसा भी आएगा मेरे दोस्त, जब कामयाबी दूर खड़ी होगी और गले सिर्फ हार मिलेंगे। उस हार से सीख लेना, गलतियों को माफी देना। दर्द जब हद से गुजर जाए तो दिल से कहना, ए दिल रो ले पर तुझे टूटना नहीं है। So whatsoever happens in life, please don't forget your happiness is eagerly waiting on the other side. Don't let it down. Keep going and keep going. Thank you very much. And in continuation of my poem, I have a dance performance depicting the life of a PhD student. I hope you like it. Thank you.
आप डांस परफॉर्म
तेरे बिन अब रह नहीं सकते, तेरे बिना क्या वजूद मेरा, तुझसे जुदा अगर हो। हम तेरे बिन अब रह नहीं सकते, तेरे बिना क्या वजूद मेरा, तुझसे जुदा अगर हो जाएंगे तो खुद से ही हो जाएंगे जुदा, क्योंकि तुम ही हो, अब तुम ही हो जिंदगी, अब तुम ही हो चैन भी, मेरा दर्द भी, मेरी आशिकी, अब तुम ही हो। तुम ही हो जिंदगी, अब तुम ही हो चैन भी, मेरा दर्द भी, मेरी आशिकी, अब तुम ही हो। तेरे लिए ही जिया मैं खुद को जो दे दिया है, तेरी वफा ने मुझको संभाला, सारे गमों को दिल से निकाला, तेरे साथ मेरा है नसीब जुड़ा, तुझे पाके अधूरा न रहा, क्योंकि तुम ही हो, अब तुम ही हो जिंदगी, अब तुम ही हो चैन भी, मेरा दर्द भी, मेरी आशिकी।
Thanks, Zora, for such a dynamic performance. Now I would like to call on stage every member of the organizing committee of entertainment program as well as every participant.
So honestly, there is nothing to say, right? Give them a big hand. These students have done really something which I never expected, right? I was thinking, okay, this is going to be something, okay, but they've just floored it, right? So in addition to the solo performances, which went excellent, the skit was something which was amazing, right? The entire troupe was put together, the script, everything was put together by Rohit and Sneha. All the committee did was, okay, you need to do this, come at this time, make sure that you are here, so on and so forth, as we always do as PIs, right? But these kids really did the job. So give them again a big hand.
I would like to just put one word. Rajni was also very important here because she really understood what they want, and in fact, I think provided some samosas also to everyone, not only the finance officer, during the rehearsals. Yes, and so give a big hand for Rajni also here.
So the admin, the IT, and and Rajni and Yogita, Pallavi, everybody in the committee did an excellent job. So hands to them.
So they came to me after I think 4 days of rehearsal. Ma'am, बहुत मुश्किल हो रही है entertainment के लिए, बंदे नहीं मिल रहे. We need support staff. So I would also please want you to clap for the support staff who really came at the last moment, and that was really wanted backstage. Thank you.
Thank you so much. Now, I now have the pleasure of inviting Professor Richard Hans, who is here with us online to deliver the State of the Art Foundation Day Scientific Lecture, Therapeutics for SARS-CoV-2 and Beyond, the Recovery Trial. Professor Richard Hans, there has preclinical. May I please ask the audience to please be seated? Professor Richard Hans, there has preclinical medical studies in Cambridge before moving to Oxford for his clinical studies and qualified in 2000. He came to CTSU for a period of out-of-program reach in 2006 to work on the HPS-2 TRIVE trial with Professor Jane Armitage. He was appointed to the MRC Program Leader track in 2011 and is now a program leader in the MRC Population Health Research Unit for the program in randomized trials and cardiovascular and metabolic disease. I would request our team to please support us for the talk. I welcome you, sir, on the behalf of the THSI members.
Can you hear me okay?
Yes, sir, we can hear you.
And you can see my slides?
We are able to see you right now.
Can you see my slide?
Not yet.
Okay, I'm sharing my screen, so you should be able to see my slides.
I think in a few seconds we will be able to. Yes, we are able to see your slides right now.
Okay, great. Well, thank you very much for the introduction. I'm sorry that you didn't get Professor Sir Peter Horby today. Unfortunately, he's been called away on urgent business, but I've had the pleasure of working with him on the recovery trial for the past 2 years. So COVID-19, as we all know, has been a disease which we've all learned a huge amount about over the last 2 years. And it's a disease which affords clinical trials multiple opportunities to improve the outcome. So in most people, COVID-19 begins as a viral illness and resolves as such, and most people don't get particularly unwell with it. Some patients do become more unwell and they develop pneumonia, which is stage 2 on the figure I'm showing you here, and they may require hospitalization. And then a subset of those patients then develop a exaggerated host inflammatory response. And those patients are the ones that become most critically unwell and may frequently require mechanical ventilation or other organ support. The advantage this gives us as clinical trialists is we have a disease which has multiple different stages which we can target to try and improve outcomes. So the recovery trial was set up in early March 2020 by two professors at the University of Oxford, one Peter Horby and one Martin Landray. They actually effectively met on this bus, which is the number 18 bus, which runs in London from one of the train stations to somewhere where they work. And they met on this bus just by chance and were discussing the outbreak of COVID-19 that was going on in China and in North Italy. And they decided that a trial was urgently needed because it was likely that the UK was shortly going to be overwhelmed by a very large number of infections. So a team was very quickly put together at the MRC Population Health Research Unit where I work, in collaboration with people in Peter Horby's team in a different part of the university. And after the protocol was written, the first participant was then recruited 9 days later. As we all know, scientific experiments don't just rely on 1 or 2 people, but there's a whole team of people required to do a trial like Recovery. We have the chief investigators, that's Peter Horby and Martin Landry. They had a team of people around them in Oxford, including trial managers, clinical support, data managers, communications experts, IT developers and testers, and a data linkage team. And that was just the team in Oxford. And then around them, we had a site, and we had every acute hospital in the NHS was participating in this trial. And every site had its own team with a principal investigator, research staff, pharmacy, and other support staff. So Recovery was very definitely team science and a huge team effort across the UK as it got going. So I'm going to start. You should be able to see this slide now develop. And this shows each of the sites as they open. You can see the dates ticking across the top. And as the green dots get bigger, it means that the sites are recruiting more and more participants. So you can see things went very quickly in the first 100 days. So we had a team of people helping us identify the sites, get all the approvals required, get them the study drug, train the staff, and then allow them to start recruiting. And that allowed us to recruit over 10,000 patients in the first 2 months of the pandemic in the UK. But recruiting trial participants is never straightforward, and particularly not when they're very unwell and in hospital, frequently in hospital by themselves. So I'm just going to play you a video now, which is a video that we put together that participants could watch on their own phones or tablets when they heard about the trial, to help them learn some more about it. So I'll just play this, and I hope if you can't hear the sound, please let me know, but I hope you can.
The recovery trial is being carried out by the NHS and the University of Oxford and is trying to find an effective treatment for COVID-19. You or your child are being invited to take part in the trial because you've been admitted to hospital and have, or are suspected to have, COVID-19. We're testing several treatments suggested by the expert panel that advises the Chief Medical Officer in England. We don't know if they'll be effective, but experts think they should be tested in people of all ages. So this treatment may or may not help you or your child personally, but this study should help future patients. The possible side effects of these treatments are well known for other uses, so your doctor will know what to look out for and can discuss this with you. You can also find more details in the full information sheet you've been given. If you or your child join the study, we will need a signed consent form and some general health information. Then you'll be randomly allocated to one of the treatment options by a computer. You will either receive the usual standard of care that all patients receive with no additional treatment, or the usual standard of care plus one of the treatments, which might be given by mouth, injection, or inhalation. Neither you nor your doctor can choose which treatment you'll receive, but patients will only be included if their doctor thinks they're suitable. During the trial, we will record patient health information which will be stored confidentially and securely on computers at the University of Oxford. We will get this information from your medical records. When you leave the hospital, there will be no further visits for the trial, but we may in some cases access your medical data held by the NHS for up to 10 years. Joining the study is voluntary, and you or your doctor can stop the treatment at any time. If you want any more information about the recovery trial, please talk to one of the hospital medical team. We will be happy to answer your questions. Thank you for considering this trial.
So that video has been watched many thousands of times by potential participants and helped us to recruit the large numbers that we have. These are some baseline characteristics of the sorts of people that we've been recruiting into the trial, which match very well with the sorts of people admitted to hospital with COVID-19. So they're older patients, average age of 63, nearly 2/3 are male. At the time this data was cut, this was almost entirely a UK population, 75% of whom are white. They'd been unwell for about 9 days. And you can see that some people were in hospital but did not require oxygen. The vast majority of people were in hospital receiving oxygen, and 10% of people were in hospital actually on a mechanical ventilator at the time that they were entered into the trial. But over half of people had a major comorbidity, something like diabetes or heart disease. So this was, as I say, fairly typical of the population of people being admitted to hospital around this time. So our first results came out much more quickly than we were expecting. And these were results of when we were testing a drug called hydroxychloroquine. And actually, this was stopped slightly early because an independent group of experts, the Data Monitoring Committee, advised the steering committee that the drug was not going to be found to be beneficial. And you can see on this Kaplan-Meier curve showing the number of days after randomization and the proportion of patients who died, that in the usual care arm, that was about 25% of people who randomized had died within 28 days. People given hydroxychloroquine, if anything, that proportion was anything a bit higher, not significantly higher, but the committee decided that there was no chance of hydroxychloroquine being proven to be beneficial, so this comparison was stopped. And this led to some rapid changes in regulatory and clinical guidelines. The FDA, the American drug regulator who had approved hydroxychloroquine for emergency use, then revoked their emergency use authorization. And this happened on the 15th of June, which was just 86 days after the trial started. So very quick results, causing real changes in clinical practice. Not long after, we then presented the dexamethasone results. This was quite different. In this, we demonstrated that dexamethasone could save lives. For people who were on invasive mechanical ventilation at baseline, dexamethasone reduced the risk of death by about 1/3, meaning that if we were to treat 8 patients, instead of 5 of them dying, only 4 would die, which was a huge step forward, given that there were no other treatments for COVID-19 known at that time, and there was no even prospect of a vaccine when these results came out. So this was very exciting news. For patients who were ill enough to require oxygen, there was still benefit, a reduction in the risk of death by about 1/5. But for patients who were not on oxygen, dexamethasone was not recommended. This led to immediate changes in practice in the UK, and with guidance being released the same day, but around the world, practice changed after that. And that result came out 87 days into the pandemic. We found some other important negative results for drugs which many experts thought could be beneficial. So lopinavir is an antiretroviral treatment, thought to have activity against the virus, but it had no effect on mortality. Similarly, azithromycin is a common antibiotic, but thought to have immunomodulatory properties, but was found to have no benefit. Convalescent plasma was an important treatment, which many hundreds of millions of dollars were spent in the US setting up a convalescent plasma program. In the UK, we randomized over 11,000 people to receive either convalescent plasma or usual care. And we showed very clearly that it had no benefit. And therefore, these convalescent plasma programs were closed down, not only in the UK, but also in India, which allowed important resources to be diverted elsewhere. We tested other treatments which people thought could work, and colchicine and aspirin were good candidates because they are readily affordable and widely available, but unfortunately, they didn't show any clear benefits on mortality either. A later result was an important proof of principle to show that antiviral therapies can work in hospitalized patients. There was dogma at the time that once patients were in hospital, antiviral therapies would no longer be of any benefit. The virus was no longer important and we should only focus on the exaggerated immune response that I showed you earlier. However, we tested in recovery a combination of 2 monoclonal antibodies in a combination called Ronapreve in the UK. But these antibodies have been selected because they bound to the SARS-CoV-2 spike protein and were capable of neutralizing the virus. And what we found here was some very interesting results. It was already known that this antibody combination could reduce the risk of hospitalization or death among outpatients with COVID-19. So early in disease, you could stop people's disease progressing with these antibodies. But it was unknown whether giving them to people in hospital would be of benefit or not. And what became clear from our results was it was critically important to know whether people had already generated their own antibody response against the virus or not at the point they were randomized. Some patients had not. They were called seronegative patients, so they had no detectable antibodies despite having been infected with the virus about 9 days earlier. They still had no antibodies. And you can see in this seronegative population, if you just look at the blue lines here, you can see in the seronegative population, the risk of death within 28 days was about 30%, so very high. By contrast, people who had already developed their own antibodies, what we call the seropositive group, their risk of death was much lower, about half of that of the seronegative group. But not only did whether you had antibodies or not predict your prognosis with COVID-19, it also appeared to determine whether you responded to this treatment or not. So if you already had antibodies, giving you more in the form of this treatment called Ronapreve was of no benefit. By contrast, if you did not have your own antibodies, then giving you some reduced your risk of death by about 1/5, a very highly significant result. And this is really important. Although, unfortunately, these antibodies are not active against many of the Omicron variants, it does show that antiviral treatments can work in hospitalized patients. So it's important that we continue to test them and that we don't just accept the dogma that antiviral treatments will not work in hospitalized patients. And so recovery celebrated its 2-year anniversary not very long ago. And this slide just summarizes where we're up to with the trial. We have now found 4 effective treatments. So in addition to dexamethasone and the monoclonal antibodies, the Ronapreve I've just told you about, we've also found that a different monoclonal antibody called tocilizumab, which is a powerful anti-inflammatory, can also reduce the risk of death. And we've also found that more lately, a baricitinib, another anti-inflammatory treatment, can further reduce the risk of death. So we've now found 4 effective treatments after 2 years of recovery. And I think recovery shows what can happen when an academic trial works. Unfortunately, in COVID-19, there has been a lot of academic activity which has not been helpful. And this is an analysis done by some people who work at the American drug regulator, the FDA, showing the number of different trials that have been started during the COVID-19 pandemic. And you can see that of the patients who've been recruited into these trials, 500,000 people, only a fraction, unfortunately, were of trials that were adequately powered. So a tiny proportion of the comparisons being tested, just 5%, 1 in 20, had any opportunity to find a result which could inform clinical practice. The vast majority of trials which have been done have not been informative, which is a real shame. But I hope that recovery has shown that this can be turned around. So the trial is now not only in the UK. COVID-19 is a pandemic, so it needs global solutions. And you can see on this map the other countries that are now participating in recovery, including, I'm delighted to say, India. And this means that the trial can hopefully continue to recruit. Even if the pandemic is relatively quiet in one part of the world, the chances are that in another part, there will be disease activity and the trial can continue to recruit. So we're currently testing a number of treatments. Some are directed at the host, the inflammatory response. These include high-dose dexamethasone, comparing it with the standard dose that I've already shown you the results for. And we're testing a drug called empagliflozin, which is actually a drug developed for diabetes, but for which there is reason to think that it may have some benefits in COVID-19. And we're also testing novel antiviral therapies that are being developed specifically for COVID-19. So following on from the Ronapreve research, which I've shown you, we're testing another monoclonal antibody, which does have activity against Omicron, called sotrovimab. And we're also testing two small molecule oral antiviral therapies, molnupiravir and nirmatrelvir, better known as Paxlovid. These two have been shown to work in outpatients, but we're giving them a proper test amongst inpatients. But we're not stopping there with recovery. Although we've done a lot of work in COVID-19, it's actually a very good platform for assessing other respiratory infections. And an important one around the world and in the UK is influenza. It can cause between 10 and 20,000 deaths per year in the UK, but we have no proven therapies for severe influenza. So this is ideally suited for a trial very much like recovery. And we know that we have a busy period coming up. So these are data from Australia. And in the red line, you can see the number of cases of flu that have been reported during their current winter. So this is going on now, and you can see there's been a steep rise in the number of cases, far exceeding those of any of the previous 5 years. And what happens in Australia in their winter going on now is usually a good indicator of what the winter in the UK to follow will be like. So we anticipate that there will be a large amount of flu around in the UK when we come to our next winter. And this is likely to coincide with a further wave of infections of COVID-19 during the winter. So we unfortunately anticipate a very difficult winter coming up with multiple respiratory viral infections circulating. And so we're therefore very keen that recovery continues and is able to assess not only treatments for COVID-19, which will continue to be a problem, but also treatments for influenza. So these are the drugs that we're testing for influenza. Oseltamivir, otherwise known as Tamiflu, you may have heard of. It's a well-established antiviral medication, but actually there's no reliable evidence that it improves outcomes in severe influenza. We're also testing a novel antiviral, another small molecule called baloxavir. And then finally, we will test steroids again. In fact, the dexamethasone protocol that we assessed in recovery for COVID-19 was based upon a protocol which had been written for epidemic influenza. So in fact, we're going back to the start and testing corticosteroids amongst people with influenza. We've learned a lot from recovery. And these are just some of the lessons. We've learned that you can integrate high quality research into routine clinical care and provide benefits for both. The NHS was under such strain when COVID-19 pandemic was raging in the UK that many people thought it simply wasn't possible to do research at the same time. So we made great efforts in recovery to make it as simple as possible so that the doctors and nurses working with patients with COVID-19, caring for them, could at the same time recruit them into a trial. This obviously had benefits for the trial, but it also had benefits for the patient care in that it focused efforts on treatments which might work, and we're testing them reliably and systematically, rather than people being prescribed treatments for no particular good reason. So there were benefits to both sides by integrating the trial into routine care. We also made the trial as transparent as possible. So all of the trial documents that have ever been sent to an ethics committee, all of the letters we've ever had from our data monitoring committee are publicly available on the website, so that anybody can find out what's going on and what the trial is doing. It's been a huge collaboration. The local investigators are included in all of our authorship groups. and all contributing staff, over 7,000 of them, that's people who've done any form of training on recovery, will also be indexed on PubMed in the relevant publications. So it's been fantastic to be able to acknowledge the huge amounts of work that has gone in across the country and indeed now across the world into making this trial a reality. And we hope that the lessons from recovery can be applied not just to COVID-19 or not just to other infectious diseases like flu, but we hope that some of the methods and lessons that we've learned can be applied to trials in many other conditions, acute and chronic, all over the world, to make sure that trials really can transform the care that we give for all of the different diseases that burden our public health. So thank you very much for your attention. I would just like to finish by thanking everybody who funded us and helped work with us to get this trial started. In particular, I'd like to thank all of the doctors, research nurses, pharmacists, and other support staff in all of the participating hospitals in the UK and abroad. But my final and biggest vote of thanks, most important vote of thanks, must go to the participants, those patients who were willing to enter a trial at a very terrifying time of their life, and by doing so, hopefully help themselves, but undoubtedly helped health systems around the world improve the care that they give for COVID-19. So thank you very much.
Thank you, Dr. Hens, for your impressive talk and especially taking time out of your busy schedule. Our director, Professor Garg, would like to say a few words.
Thank you very much, Professor Haynes, for a very, very illuminating talk on the recovery trial. The reason we requested for this kind of a talk is that we realize this trial has actually saved hundreds and thousands of lives all over the world. And to my mind, if one Nobel Prize has to be given, it should be given to the trialist of the recovery trial. There's no doubt in my mind. So thank you very much. Can you hear the big round of applause here for the recovery trial? Because people don't realize the importance of the trial done at a time when people were dying all over the world. And to conduct a trial at such a large scale and so many sites, 177 sites, it's amazing, and including more than 40,000 patients during the pandemic. I mean, all the crisis there was I would say, out of the normal minds thinking that at least something of this sort can be done. So congratulations and compliments to the entire team from our side, from all over India. I will compliment you and the leaders of the recovery trial, and please convey our best wishes. If there are any particular question from the audience, Professor Haynes might be able to take that question. So one question I have, Richard, is that when a patient is on oxygen and or patient is not able to take consent, did you take consent from the relatives? Because often the problem in India we faced was that taking consent from hospitalized patients was rather difficult. And that's why, many patients couldn't be included. So your thoughts on that?
So I think the laws around this vary around the world. In the UK, if the patient isn't able to give consent for themselves, the relatives don't give consent. They can be consulted and asked for what they think would be their relatives' best, what they would want to do. But actually, if they can't give consent, then an independent doctor can give consent on their behalf. So that's the process we had to use in those situations.
Right. And how difficult was it for you to get the ethical approval at 177 sites? I don't think there was a blanket approval given for all the sites. Or
in the UK, there is. In the UK, the ethics committees operate nationally. So we had one ethics committee approved, and that covered all the sites. So it made things much easier.
So I think that's a very, very important point that, otherwise become very difficult to include these many sites. So I think this is something to learn about. Any questions from anybody here? If none, Richard, we'd like to thank you once again for this very nice talk. And please convey our gratitude to Professor Harbi also. Thank you very much.
I will do. Thank you very much. Thank you. Goodbye.
Thank you. Sir, Director Sir, I request Professor Girk to please come over the dais to give away the prizes for the entertainment and the quiz competition. The winners of the quiz competition, the first prize goes to Akshay and his team. The second prize goes to Dr. Bela Desai and team. And the third goes to Roshan and team. Next is the participants of entertainment program. I would like to call upon Ms. Sushmita Bakshi for the Bharatanatyam performance. I would like to invite the skit group, Ms. Monica, Mr. Rohit, Mr. Abhishek, Ms. Shruti, Mr. Shovik, Akhilesh, Sadhna, Sneh, Sunjyoti, and Pooja. Pooja, Sahil, Samriddhi, Dhyani, Mamta, Soumyadeep. Let's have a group photo with the skit group. So I would request you, all of you, to be in the dais and collect your appreciation of token. Huh? Moving to the solo performance, Mr. Abhishek Jain. Abhishek. Mr. Rohit. Rohit, I would request you to please come over the dais for collection of your token of appreciation. Shweta Pandey. Abhishek Jain for the classical performance with Rohit. Mr. Prashant. Prashant sir. Okay. Next is Ms. Shruti Lal. Mr. Shovik Chattopadhyay. Ms. Preeti Sihag. I would now like to call upon the Bhangra group, Ms. Sadhna, Sahil, Akhilesh, Mamta, and M.R. Kamlapriya. Last but not the least, the solo performance by Miss Johra Hasmi. आ गई मैम. Perfect. Thank you, sir, ma'am. हाँ, बस vote of thanks.
मैं तो देता हूँ पहले vote of thanks. No, actually, I'm kind of overwhelmed by the not only the scientific performance but also the entertainment program. Amazing talent we have, and I realize this that the entire organizing team in different sections has been working for quite some time. I don't see it, but after you see the performance like this all throughout the day, day before yesterday, you realize that how much of hard work and planning has gone into it. So thank you all very, very much. I mean, everybody included in this. And I'm truly delighted with the program. And this morning also, everybody appreciated the work being done at THSTI. It makes you actually very proud. Whatever they were saying, I mean, it was only for you, not for me. I mean, everybody knows that. So thank you all very much for this. And I hope you keep continuing the good work, which will bring your institution among the best in the country. There is no doubt in my mind. So thank you once again.
Life doesn't require us that we be the best. It tells that only we need to try our best. So let's try our best for success and to be efficient as possible. With this, I request Dr. Krishna Mohan Atmakuri to come over to the podium once again to deliver the vote of thanks.
I take this opportunity to first thank Dr. Richard Haynes for joining us in delivering the exciting talk on the therapeutics for SARS-CoV-2 and beyond the recovery trial. I should thank all of you who are in the audience who enjoyed this complete celebration right from past 2 days. So thank you all. I thank the administration which was behind the entire thing and which came in different pieces and parts and all the kind of services they have rendered as quickly as possible. If this could continue through the entire year in terms of how we get the funding, how we get everything done, that will be fantastic. And I thank all the organizing committees in making the Foundation Day a grand success. I think without the organizing committees, it would have been very difficult to get this done in such a short period of time, given the other kind of work which we normally do. Right? I thank all the students, the participants of each of the programs, the research staff and the administration staff, the security, the IT, every single person who has contributed in some way or the other towards this. And thanks to all of them. All the posters were great. The quiz was very entertaining. The entertainment program just floored the house. And thank you all for that. I take this opportunity to thank the director, Dr. Garg, for making this happen. Thank you very much.
I would now request all of you to please stand up for the Jana Gana Mana.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष
आशीष मांगे, गाहे तब जय गाथा। जन गण मंगल दायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
Thank you so much, everyone. I would request you to join us for the high tea. Thank you.